কাজের হিসেব সারা সারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার মতো, হলো কাজের হিসেব সারা। হেথা নাই গো মৃত্যু নাই কো যারা, বলে আয় রে ছুটে আয় রে তরা। হেথা নাই কো মৃত্যু নাই কো যারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার মতো, হলো কাজের হিসেব সারা। হেথা নাই গো মৃত্যু নাই কো যারা, বলে আয় রে ছুটে আয় রে তরা। হেথা নাই কো মৃত্যু নাই কো যারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার মতো, হলো কাজের হিসেব সারা। নাইকো যারা বলে আয় রে ছুটে আয় রে তরা, হেথা নাইকো মৃত্যু নাইকো যারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার মতো, হলো কাজের হিসেব সারা সারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথের পাশে ছোটে হাওয়া পাগল পারা। এত আনন্দ আয়োজন সবই বৃথা আমায় ছাড়া। ভরে থাকুক আমার মুঠো, দুই চোখে থাকুক ধারা। এলো সময় রাজার মতো, হলো কাজের হিসেব সারা।
হ্যালো,
শোনা যাচ্ছে তো?
হ্যাঁ শোনা যাচ্ছে। শোনা যাচ্ছে? হ্যাঁ
হ্যাঁ হ্যাঁ যাচ্ছে।
আচ্ছা আচ্ছা, ঠিক আছে ঠিক আছে।
বলে আয় রে ছুটে আয় রে তারা, হেথা নাই গো
হ্যালো,
নাই গো যারা। বলে আয় রে ছুটে,
হ্যাঁ শোনা যাচ্ছে। হ্যাঁ
আয় রে তারা, হেথা নাই গো মৃত্যু, নাই গো যারা। দেখো আলোয় আলো আকাশ, দেখো আকাশ তারায় ভরা, দেখো যাওয়ার পথে পাশে
হ্যালো
ছোট হাওয়া পাগল।
হ্যালো
হ্যাঁ স্যার।
এত আয়োজন সবই বৃথা আমায় ছাড়া।
হ্যাঁ স্যার, স্যার কুচবিহার কলেজে প্রোগ্রাম আছে স্যার, কাইন্ডলি জয়েন করুন। আমি স্যার বারে করেছি কিন্তু আমি কোনো সাউন্ড আসছে না আমার, ভয় পাচ্ছি কি হবে। ঠিক আছে, আমি তাহলে স্যার বলে দেবো ওদেরকে, তাই কি স্যার? বলে দিচ্ছি স্যার।
Ravindra sir, should we start?
জয়েন করা যাচ্ছে না, আর আমি জয়েন করছি বারবার, কিন্তু আমার সাউন্ড আসছে না কেন বুঝতে পারছি না।
Thank you,
Thank
sir.
you.
So, a very good evening to all of you joined today in this international webinar organized by Department of Chemistry, Cooch Behar College. We are very pleased to welcome all the dear participants and the dignitaries around the world. The topic of today's webinar is drug discovery, development and vaccination. And I think it will be one of the great webinar we may ever seen, as today we have our great resource persons from different part of India and other countries. It's an absolutely delight and honor to have you all in this evening. Thank you. We all know that the world today is facing a biggest public health risk, and a country like India, where the population density is so high, now it's become a tough challenge to overcome the pandemic. Every country is looking for a vaccine, but we all know that it's not an easy task at all. So what is the importance of this type of drugs or vaccines, and what is the process of development of vaccine? All this will be discussed today in this webinar, so please stay tuned with us. I request everyone to mute yourself and turn off your video to run the program smoothly. Thank you. Before we start, let me acknowledge the presence of our honorable and eminent guest have with us. Chief Patron of today's webinar, Dr. Dev Kumar Mukhopadhyay, Honorable Vice Chancellor, Cooch Behar Panchanan Burma University. Chief guest of the webinar, respected Dr. Abdul Kader Safeli, Honorable Registrar of Cooch Behar Panchanan Burma University. But due to some unavoidable reason, he is unable to join with us. But his blessing and wish is always with us. Chairperson of today's webinar, Dr. Cooch Kumar Devnath, Honorable Principal of Cooch Behar College. Special guest, respected Dr. Probir Kumar Haldar, Dean, Faculty of Science, Technology and Vocational Studies, Cooch Behar Panchanan Burma University. Now, first of all, I would like to request respected Dr. Robindra De, convener of today's webinar and head of the Department of Chemistry, Cooch Behar College, to welcome everyone on this auspicious webinar. Please stand.
Thank you. Thank you, Shravanti. Good evening, everyone. We, the member of organizing committee, are really feeling very happy and proud for organizing this one-day international webinar on drug discovery, development and vaccination. Firstly, I would like to warmly welcome our chief patron of the webinar, Dr. Dev Kumar Mukhopadhyay, Honorable Vice Chancellor, Cooch Behar Panchanan Burma University. Our chief guest, Dr. Abdul Kader Safeli, Registrar, Cooch Behar Panchanan Burma University. Our principal sir, Dr. Pankaj Kumar Devnath, who constantly encourages us to do different activities beside our academic activities. Our chief guest, Dr. Probir Kumar Haldar, Dean, Faculty of Science, Technology and Vocational Studies, Cooch Behar Panchanan Burma University, who delivers the keynote address of this webinar. I would like to welcome the resource persons, Dr. Anamitra Chatterjee, Regulatory Affairs Associate, Novartis, Slovenia, Dr. Ashwini Kumar, Research Associate, IIT Delhi, Dr. Goutam Biswas, Assistant Professor in Chemistry, Cooch Behar Panchanan Burma University, Dr. Arindom Chatterjee, Scientist, Web Life Science, Boston, USA. I also welcome all the teaching, non-teaching staff of our college. I warmly welcome to all the participants of our state, other states of our country, and very, very heartily welcome to the participants from abroad, USA, Kenya, Pakistan, Nepal, and so other countries who participate in this webinar. We are really very happy and very proud for them. Today's webinar is very, very relevant to the present pandemic situation, I hope all the participants will be benefited, enrich themselves from this webinar. The resource persons share their knowledge, views, working experience about the drug development, vaccination. So we really thankful to them to coming in our webinar and discuss their view, their activities. So we really very happy and proud for the resource person of today's webinar. Thank you so much. Thank you.
Okay, sir. Thank you very much. Now I would like to request respected Dr. Dev Kumar Mukhopadhyay, Honorable Vice Chancellor of Cooch Behar Kanchanan Burma University and Chief Patron of today's webinar to say something about today's webinar. Please, sir.
Thank you, Srabosti. Thank you very much. At the very outset, I express my sincere thanks to the Principal of Cooch Behar College and Head of the Department of Chemistry, Cooch Behar College, for organizing this beautiful webinar. And under such circumstances, you have chosen such a subject which is very much relevant and to the situation. And it's really a core subject of the chemistry and other medicinal science. But having background from the social science, I can say a few words regarding these things that when if you go back to the history of the invention and this discovery of quinine and the medicine of malaria, we can say that after suffering more than 100 years, what happens? It was discovered, and we know that the bark of extract of the cinchona plants was used before the discovery of this medicine for malaria. And we also have seen that from the pages of history that the fighting between the planters and the traders who used to get all these seeds, that is the specially regions in the Latin American countries like Java and other parts, that is part of the Latin American countries where it was planted and it was discovered. Then the part of the European Union, that is nowadays it is called the European Union, then Great Britain, France and other countries who used the policy of colonization, and they had to get the medicine of malaria because they have to come to the malaria-ridden provinces. That's why they have to get the medicine of malaria. Why I am talking about these things? Just for that reason, that due to the problem that was faced by Great Britain, France and other countries who took the policy of colonization, they had to get the medicine of malaria. That is that I want to say, that if we go into the pages of history, we see that when we face any problem, we face it and we try to get rid of the problem. Like that, even now also, under this situation, under COVID-19 situation, also when the total world is suffering from this problem, in every corner of the world, some research activity is there in order to get rid of this problem. Now there are two parts. If you see, what are the two parts? First part is that is the research activity is being carried out in some private institutions, and also it is being carried out in some government institutions also. What is the difference? Difference is that private industries, they have the profit motive, but with the government initiative, it is purely for the human being. So we are too dependent on the government initiative. And even in some cases, we have seen that private institutions, private traders and private industries also, they come forward with some invention, discovery also. So, under such circumstances, throughout India, throughout the world, we see on the pages of the newspaper that vaccines are under process. Some doubts are there, some controversies are there, still we are waiting for the vaccine. But two things are there, I am sure. Experts are there from different parts of the globe. They will present their thoughtful papers, deliberation, and they will speak on these subjects. But I like to just touch upon 2 or 3 points. That is why the vaccine? Why, as a person coming from social science background, what I understand that vaccine, that is, we always say that prevention is better than cure. We understand vaccine is used for the prevention of any deadly disease, and medicine is used for the cure of any disease. So it is always preferred prevention. That's why the role of vaccine is very important from the point of view of any disease or any problem we are facing in any time, any part of the world. And fortunately, in different corners of the world, research activity is going on. So we are hopeful. We are hopeful that within a very short period of time, we, the people of this world, will get some vaccine for this coronavirus. And till that time, we will have to be very much cautious. We will have to be very much careful to protect against this coronavirus. But we know that process is there for the discovery, for the invention of this vaccination and also drugs. So detailed discussion will be made by the experts coming from the discipline. And I am very much hopeful this beautiful topic that has been chosen by the Department of Chemistry, Cooch Behar College, will help teachers, students, scholars, and other participants of the webinar, and they will get some recent ideas from the persons who have come to join this webinar. On behalf of Cooch Behar Panchanan Barba University, I express my sincere thanks to all the invited speakers that they have spared some times for our purposes, and they have spared some times for the Department of Chemistry, Cooch Behar College, and especially I express my sincere thanks to Cooch Behar College for organizing a number of webinars on some recent topics that will help, that is helping also the student community, teachers, researchers, and other interested persons who are connected with this problem. Thank you very much. Thank you. Thank you. Over to Srabosti. Thank you.
Thank you, sir. Thank you very much. So, due to some unavoidable reason, our chief guest of today's webinar, respected Dr. Abdul Kader Safeli, sir, Honorable Registrar of Cooch Behar Panchanan Burma University, is unable to join us today, but we all know that his wish and his blessing is always with us. And our chairperson of today's webinar, Dr. Pankaj Kumar Devnath, Honorable Principal of Cooch Behar College, had to go out of station for important work, and he is also unable to join us today. But he left his valuable and encourageous words for us. I would like to share that clip with all of you. Give me some time.
Namaskar and good evening to one and all. It is really a pleasant moment for me to be with you all through this virtual platform of Google Meet. As the principal of Cooch Behar College, I am really proud to be associated with you and to speak on the auspicious occasion of today's webinar. Today's webinar on drug discovery, development and vaccination will definitely enlighten some of the very important issues related to drug and vaccination in the context of COVID-19. We will also able to understand the myth and realities associated with the same. I would like to convey my sincere thanks to Professor Kumar, the Honorable Vice Chancellor of Cooch Behar Panchayat University. He is not only our chief patron, but also he is the source of inspiration of all the activities of our college. We have with us the Registrar of Cooch Behar Panchayat University, Dr. Abdul Qadir Safeli, who is the chief guest of today's program. I would like to thank Dr. Safeli for his constant support and involvement with the activities and development of Cooch Behar College. We have with us Dr. Kumar, Dean Faculty of Science, Cooch Behar Panchayat University, and who will also deliver the keynote speech of today's webinar. Thank you, Dr. Haldar. We are really very thankful to all the resource persons and the eminent academicians who will speak on today's webinar as the resource persons. Though they are very busy in their academic field, still they have agreed to speak on these important issues. So I would like to thank Dr. Anamitra Chatterjee, Regulatory Affairs Associate, Novartis, Slovenia, who will speak on drug development. We have with us Dr. Ashwini Kumar, the Research Associate, IIT Delhi. He will speak on immunity and vaccines. Dr. Goutam Biswas, Associate Professor, Cooch Behar Panchayat University, will deliver his speech on nanomaterials, a possible therapeutics in SARS-CoV. And we have with us Dr. Arindom Chatterjee, scientist, Wave Life Sciences, Boston, USA, who will speak on DNA silencing in drug discovery. They are really very authority in the field of today's webinar discussions. So I would again like to thank all of them. I would like to thank all the participants of this today's webinar, and we have actually more than 200 participants from different higher education institutions, not only from our state but also from India and abroad. We are really thankful to all of you. I would like to convey my sincere thanks also to the Faculty of Science of Cooch Behar College, especially the Faculty of Department of Chemistry, who is organizing this today's webinar, Dr. Robindra De and Dr. Snehasil Gupta. They are really very doing very important works in organizing this important webinar. I would like to thank all other teachers and non-teaching staffs of Cooch Behar College, including the members of our governing body and also the president of our governing body, Shri Mihir Goswami. And lastly, I would like to conclude my speech hoping that this today's webinar will be very, very grand success and all of the participants will definitely be benefited from this today's webinar. Thank you all. Thank you.
Thank you, sir. Thank you very much. It's really meant a lot for us. Now I am requesting respected Dr. Prabir Kumar Haldar, Dean, Faculty of Science, Technology and Vocational Studies, Cooch Behar Panchanan Burma University, and special guest of today's webinar to say something regarding this webinar. Please, sir.
I am audible?
Yes, sir, absolutely.
Okay. It is a matter of great pleasure for me to be among the galaxy of faculties, students, research scholars of our beloved country and across the globe. I am thankful to the organizer for giving me an opportunity to say something as keynote speaker. On my personal and on behalf of the university, I express my gratitude to our Honorable Vice Chancellor, Professor Dev Kumar Mukhopadhyay, for accepting the invitation of the organizing committee to be the chief patron of the webinar. His profound gesture of love of the faculty and student is truly indebated. On behalf of the university, he would always lend his hand in such valuable programs. I would like to thank Dr. Pankaj Kumar Debnath, Principal of Cooch Behar College. He has arranged so many webinars to enrich the students and teachers. All of us know about the dread effect of the pandemic. It has put everything at mess, both human and non-human sphere. Everyone is waiting for new invention from the scientific world for curative measure of the coronavirus effect. We are longing for discovery of vaccine to cure this disease. But life has to run and the vehicle of education has to move. But it is understood that we cannot make teaching at far with face-to-face one, but still online education is the only way out. Therefore, we have to streamline all at our level best to move along with the need of the hour, along with the wave. In the areas like ours, where we have very low technical infrastructure, for example, internet, accessibility is low, availability of 24 hours current is not possible, and the vicinities of the college and the university are rural and remote as well. Such limitations will physically hinder the online system of education, but we should accept these challenges as an opportunity. As we know, the necessity is the mother of invention. In this period of crisis, I feel the topic of an webinar about the drug discovery, development, and vaccination is quite relevant to the present context of the education institutions. Now, I want to introduce our valuable learned resource person, Dr. Anamitra Chatterjee. He did his BSc in chemistry from Ramakrishna Mission Vidya Mandir, Bedu in 2009. After that, he finished his MSc in chemistry from IIT Bombay. Then he moved into Switzerland for PhD in 2011. After PhD, he did 2 postdocs, one from Norway and another from Germany. His research career was focused mainly on the organic chemistry. After spending so many years in Europe, his motivation was built to career in the pharma industry, mainly in Europe. So he just started his industrial career in Novartis, Slovenia as a regulatory affairs associate. We will speak about the drug development process in the pharma industry. Our second resource person Dr. Ashwini Kumar. He completed his BTech in biotechnology from the MIT University, Noida in 2008 and MTech in biotechnology from National Institute of Technology, Rourkela in 2010. And his broad area of research work was developing mucoadhesive drug delivery system. And he joined Department of Biochemical Engineering and Biotechnology, IIT Delhi, as a research associate in March 2020. And Dr. Goutam Biswas, assistant professor of Cooch Behar, Burma University. He is presently pursuing research towards synthesis of the small molecular having therapeutic activities in nanomaterial. He obtained his PhD from IICB, that is Indian Institute of Chemical Biology, India, in 2006. And he has over 15 years of experience in different fields of chemistry and total synthesis methodology, organometallic solid phase synthesis, and also of nanochemistry. He has a co-author of 16 scientific publications and 4 patents. His research interests include carbohydrate, chemical, nanomaterial, synthesis, and medical. In spite of those, he is an excellent teacher. He is dedicated to our university and dedicated to the students. And in spite of this corona age, he is pursuing his research work in every day. So, next resource person, our Dr. Arindom Chatterjee. He received his undergraduate with chemistry honors from Ramakrishna Mission Vidya Mandir, Belur Math, under University of Calcutta in 2009, and doing his masters in organic chemistry from IIT Madras. After completing masters, he joined University of Connecticut, USA, as a graduate student of PhD under supervision of Professor Ashwini Kumar. His PhD research was mainly focused on the synthesis of the site-specific DNA adducts of the aromatic amines and nitroaromatic carcinogens and study of their replication properties. After finishing PhD, he joined Web Life Science, Lexington, USA. That is a biotechnology company working on developing new for rare genetic disease as an oligonucleotide process scientist. So I once again thank all the esteemed speakers for participation in the webinar. Lastly, I would like to thank Principal Dr. Pankaj Devnath, an organizer of the webinar. I especially thanks the head of the Department of Chemistry, Dr. Robindra De, to arrange such a beautiful program. I wish everyone all the best for their endeavors. Thank you all.
Thank you very much, sir. Now we are going to start our main session. Before that, I want to make an announcement that if you want to ask any question to our resource persons during the session, then please feel free to ask and write your question in the chat box. We will definitely try to pick up your question from chat box after the completion of respective session. So, today, our first resource person of today's webinar is respected Dr. Anamitra Chatterjee. Dr. Chatterjee is from Bankura, West Bengal. He did his BSc in chemistry from Ramakrishna Mission Vidya Mandir, Belur in 2009 and MSc in chemistry from IIT Bombay. He did his PhD from Switzerland and 2 postdoctorate from Norway and Germany. His research career was focused mainly in organic chemistry and however in diverse fields, namely artificial metalloenzymes, photoredox catalysts, etc. After spending 8 years in Europe, his motivation was to build a career in the pharma industry, mainly in Europe. So he just started his industrial career in Novartis, Slovenia, as a regulatory affairs associate. Today, he will speak about the drug development process in the pharma industry. So now, I would like to request Dr. Anamitra Chatterjee to take over the microphone and start with his presentation. Please, sir.
Thank you, Saranti, for your kind introduction. Just let me know when you can see my screen.
Yes, sir.
Perfect. So thank you, Saranti, and thank you also, Mr. Pravir Halder, for your kind introduction. And I also would like to thank for the invitation for giving me such opportunity for the presentation. I really appreciate the Cooch Behar College and also the HOD, Dr. Robindo sir, and also to all organizers to choose really such highly relevant topic with the current situation. I really hope that all the topics will be interesting, and I'm really looking forward all the lectures that will become also in later. So without delaying, I would like to start with my presentation. So I would like to talk about the drug discovery and the development. So this slide actually presents the roadmap of the drug discovery and the development. So you can see there are several stages that involves the drug discovery and development. So for example, the drug discovery stage, preclinical stage, clinical trial stage, and then the health authority review, and then it comes for marketing the drug in the market. If you look to the timeline, that drug discovery and preclinical stage takes roughly about 3 to 6 years. And then it comes for clinical trials, which takes roughly 6 to 7 years. And then it comes for health authority review, which takes roughly 1 to 2 years. So the whole procedures from start to end, so from discovery to launch to the market, it takes roughly 12 to 15 years to develop a drug compound. And if you look to the picture carefully regarding the molecules or the compounds that we suspect as a drug, so at the beginning, there are 5 to 10,000 of compounds, and then it decreases to 250 compounds in the preclinical stage, and then in clinical trials, it comes 5 to 10 compounds, and then finally one compound approved as a drug. So you can see that this is a very long procedure, and it has a lot of risk in the drug development process. So in next couple of slides, I will take you through the whole roadmap step by step. So I divided the whole procedures in the 4 steps. So drug discovery, and then preclinical study or the animal studies, and then the clinical phase or clinical test, and then the commercialization. So let's start with the drug discovery phase. So when in the drug discovery, you choose a particular disease that you want to cure basically. And for that particular disease, you have to choose a drug target. So drug target is a biological molecule. And then you identify a bioassay. The bioassay is a test that is used to determine the biological activity. So I would like to talk about the drug target. Drug target is really important. It's a kind of specific macromolecule of the biological system that actually interact with the drug. So drug has to be really specified with the drug target to cure the particular disease. I would like to give a simple example here. For example, the serotonin. So we are talking about the depression. So we know the depression comes when the serotonin uptake happens. So serotonin is a neurotransmitter. And classically, this tricyclic compound is known to inhibit the serotonin uptake. And in the pharma company actually developed several molecules to screen for how you can inhibit the serotonin uptake. So then they found that Prozac compound can be suitable to inhibit the serotonin uptake. So I'm just giving a simple example here. So for example, serotonin here is the drug target and the Prozac compound is the drug compound. So just to understand very simple what is the drug target and the drug compound. And then having the genetic code for the production of an enzyme or the receptor that may enable us to overexpress the protein or determine its structure for the biological function. And if seems to be really important to disease process or inhibitors or the antagonist can be prepared through the process that is called rational drug design. Also, simultaneously, our chemistry is improving. So, for example, nowadays, the combinatorial chemistry can be used for large number of compound synthesis, and it really takes less time. And also the efficiency of the synthetic chemical transformation is really improving with upcoming the new technologies. The drug selectivity is really important because when you are targeting for particular enzyme or particular biological function, the selectivity will be really, really important. Otherwise, your drug can have several side effects. And this kind of study is much more known about the biological chemistry of the living system. So, for example, the targeting one subtype of the receptor that may enable the pharmaceutical chemist to avoid different side effects associated with the drugs. And of course, this study can involve a lot of side effects or a lot of problems. So, for example, you choose a target and maybe over time, it can lose its sensitivity to the drug. So, for example, it happens that penicillin binding protein, which is known to the primary target of penicillin in the bacterial species Staphylococcus, has evolved a mutant form that no longer recognizes the penicillin anymore. So this kind of problem, of course, is associated with this kind of study. So target is to find a lead compound. What is the lead compound? The lead compound is the structure that has some activity against the chosen target, but not yet good enough to be a drug itself. And if it is not known, then you have to determine the structure of this lead compound. And normally the lead compound can be found with the high throughput screening, that is, that technology are used in the pharma company. So pharma company actually synthesize thousands of compounds. And you can see, of course, in this picture that there are thousands of compounds are synthesized, and this can be stored, cataloged, and then you screen for particular new targets for the disease to find the lead compound. And when you find the lead compound, then the toxicological study is really important. And the toxicity study is done with different QSAR models nowadays available. And the QSAR is basically the structure-activity relationship. So you have to see that your lead compound is mutagenic or not, if it is a toxic to your health or not. So this kind of study you have to done afterwards. So the goal is to identify the pharmacophore. The pharmacophore is the structural features that directly responsible for the biological activity. And when you find the pharmacophore, for example, you can see this is a pharmacophore. When you find a pharmacophore, you have to optimize the structure to improve the interaction with the biological target. So for example, this is the pharmacophore, and now you have to play with different chemical technology. So for example, you can modify some functional groups that are involved such way that it can interact very nicely with surrounding amino acid or with surrounding biological system. This is a computer-assisted drug design. So if one knows a precise molecular structure of a particular biological target, for example, enzyme or the receptor, then one can use a computer to design a perfectly fitting ligand. But of course, there is several drawbacks. For example, the commercially available programs doesn't allow the conformational movement in the particular biological system. So these programs are somehow inaccurate representation of the reality. But this is not our less hope. I hope in the future, much more software will be designed and it will be solved, this problem, the computer-assisted drug design. Of course, serendipity is a different issue. So it is associated with the experimentalist who has much more experience and who understands the big picture of the drug development, and also who has an open mind toward the unexpected results and also can use the deductive logic in the explanation of such results. So for example, the penicillin discovery or the development of Viagra is a kind of serendipity example. Okay, now we talked about the drug discovery to find the lead compound, to find the pharmacophore, and to find some biological activity of the chemical compounds. Now, when we have this kind of compounds, we have to do the animal study. Why we perform the animal study? Just to find the toxicity and the efficacy in the animal models. And we also want to know how the drug actually do the mechanism in our body. But this kind of study is not so easy because this involves lot of animals, lot of screening, and each animal is slightly different with different metabolic systems. So one drug may be toxic to one species, but not to the another. We also determine the pharmacodynamics and the pharmacokinetics of the drug here. The pharmacodynamics actually explores what a drug does to the body, and the pharmacokinetics explores what the body does to the drug. Because when you take a drug to your body, this drug actually is a foreign substance to your body. So your body doesn't like it, I will say. So these kind of foreign substance are referred as a xenobiotics. So body has a goal to remove such xenobiotics from the system by excretion in the urine. And your kidney is designed such a way that it allows a polar substance to escape in the urine. So body has a tendency to transform this drug compound to more polar structure, so such that it can remove from your body. A particular enzyme is involved here. It is called cytochrome P450, which actually convert a non-polar bonds, which is, for example, RH or CH, to a more polar bonds to ROH in a hydroxyl group. So it can easily remove through the urine. But when you are doing such kind of study, you always have to file a patent because there are several companies involved in the drug development market and you want to avoid the competition. So you file the patent and then you continue to study the drug metabolism and then you continue to study the toxicity. But this kind of in vivo test or the preclinical study is really complicated because of the really complicated biological system, and this can be interfered with several biological processes which is maybe unknown to scientists. So this kind of study is really expensive and it causes suffering to the animals. So when we perform the animal study or the preclinical study, we have some information on the mechanism of the drug. We have some information on the toxicological study. So now we would like to perform the clinical study. But before you do the clinical study, you have to perform the manufacturing process. You want to produce the drug in the large amount. So when you are going to produce the large scale amount of the drug, the pharma company always makes profit. So we always have to be careful how we can sell. The drugs must be sold at a profit. So profit, when you are looking for profit, you are thinking about the cost effective route of synthesis of the drug. So one must be very careful in designing the route of synthesis, which can involve actually a readily available or inexpensive starting materials, for example. Or the route of synthesis can be as few steps as possible or inexpensive reagents involved in that route of synthesis. Then also your route of synthesis must be suitable to scale up because you want to produce in the large scale and you have to produce at least 10 kilograms of the final product. And when you are designing such cost-effective route of synthesis of the drug, you also can consider, depending on the microbial process, instead of the synthetic methodology, to synthesize some intermediate. So then your drug will be the semi-synthetic product. So these are the cost-effective technology that pharma company normally use. So when we have large scale amount of the drug, now we would like to do the clinical trials. So directly apply to the human body. So the first stage is the phase 1 of the clinical trial. And in phase 1 clinical trials, we normally choose 20 to 80 healthy volunteers to apply our drug. And drug is tested to determine the toxicity that is relative to the dose and to screen what kind of side effects can happen. So then after phase 1, we jump for phase 2 clinical trials. And here much more volunteers are involved. Normally it is 100 of participants, I will say. And the goal of this phase 2 clinical trial is to determine the beneficial effect of the drug and to determine the dose level that need for this kind of effect. When we done the phase 2 clinical trial, then we move for the phase 3 clinical trial. In phase 3 clinical trial, normally 100 to 1,000 of participants are involved. And the goal of the phase 3 trial is to determine the safety and efficacy, both. So we compare the existing treatment with a placebo. So placebo, if you don't know, placebo is the drug compound, but without any active agent, without any API. So that the API is the main component that needs to cure your disease. So with placebo, you should not expect any kind of beneficial effect with your disease. Then if phase 3 clinical trial is successful, usually the regulatory affairs department in the pharma submit for the new drug application to the health authority. Then health authority review all the data from the manufacturing, how this drug actually manufactures, how you perform the clinical study, preclinical study, every documents they check. And then after checking everything, if they found that drug is okay, then they can approve or they can reject it. If suppose they approve, then you are ready to launch in the market. Then it comes to the phase 4 clinical trial. In the phase 4 clinical trial, there are thousands of participants are involved. And the purpose of the phase 4 clinical trial is to study the long-term side effects or long-term benefits in the human body. So what about the total cost that involved from the drug discovery to up to clinical trials and then the marketing to the marketing of the drug? So I will say it is a really difficult questions to answer, but I can give you a rough estimation. Nowadays, to develop a drug, it costs roughly $500 million to $2 billion US. And this is a breakdown of the cost. You can see the most money involved in the discovery phase and in the clinical trials phase. So if you go to the pharmacy and if you want to buy a brand drug, it costs a little bit much more amount of money, but maybe the pharmacist can offer you the generic version of the drug, which costs really less price of the same drug. So you will be surprised, why is that? Because generic drugs actually doesn't involve such huge clinical study that is involved in the branded drug. And also it doesn't involve the discovery and the screening phase. So it actually saves a lot of time to develop the generic drug. And also it saves a lot of much amount of money that invest to develop the clinical study and the discovery phase. So that's why generic drugs actually sells in very less price compared to a brand drug. And now I would like to say a little bit about the drug regulatory bodies. So drug regulatory bodies are the organization that actually allow that a drug will be marketed or not. So if a pharma company develops a drug and want to launch in the market, so they need a permission from the different regulatory bodies. So for example, if any pharma company wants to launch a drug compound in the US, they need approval from the FDA. If any company wants to launch in the market in Europe, they need approval from EMA. And in Japan, this organization called PMDA. In UK, it is called MHRA. And in India, it is called CDSCO, the Central Drug Standard Control Organization. And these different regulatory bodies has different rules and regulation in different countries. So that's why the ICH, the International Conference of Harmonization, has a goal to harmonize these regulatory guidelines so that all the times that it takes for approval of the drug, it can be very short. So patient can easily avail the drug products in very short time. So that brings to end of my presentation. Thank you very much for your kind attention. And if you have any question, please feel free to ask. Thanks.
Thank you, sir. It was really a great session. Thank you. And we have got some questions in our chat box. So first question from Dr. Tapun Kumar Das. How much time to require to invent anti-COVID drug or immunization of COVID-19? As you told, it is a procedure of at least 12 years.
Yeah, exactly. So this is the usual procedure that it takes 12 to 15 years, but this is a COVID situation and this is a pandemic situation. So pharma company are really trying hard to reduce the time that is involved. So we cannot expect that the vaccine will come after 12 years, but but the clinical study and the preclinical study, it takes really lot of time. So the drug development stage also can be shorter, but how one can really sort the clinical study, that is, so you want to apply the drug to the human body and you want to observe if any kind of side effects happening or not. So I will say this is really challenging for a pharma company to reduce the time from 12 years to 1 or 2 years. But of course, one can expect that it will be solved very soon. But I think many pharma companies are trying really hard. Next question, I will say.
So next question from Ritik Pal. In which basis we choose an animal for test?
Usually, the basis is for toxicological study. So when you develop a drug and you don't want to apply directly to the human body because this drug has many side effects. So first you want to try in the animal just to understand the mechanism of the drug and what kind of side effects can happen. It can kill the human cell or the mammal cell, basically. So that kind of study you want to do in animal first, and then you come step by step to the human body. So that is the basis, I will say.
The third question from Lovely Gupta is whether repurposing of drug is patentable or not, like in case of COVID-19.
Yeah, of course it is patentable. I think it depends. I think company will be patent first.
Okay, sir. Next question. Why do some vaccine require boosters?
I'm not vaccine specialist, so maybe the next talk will be on vaccination or vaccine. I will suggest maybe this question can be asked to the next person.
Okay, sir. Next question. Why is the new corona vaccine discovered so fast in few months and not years? Do they not follow the actual protocol and phases to discover a drug?
I know the corona case. I know about my company. My company actually use a malaria the drug to cure the vaccine. the hydrochloroquine or something like that. So these are kind of not a new compound or new molecule that takes a long time to discover. So it is a kind of known compound, and then they tried directly to the preclinical study or clinical study. That's why it takes really few months. But if you really want to develop a completely new entity or new compound, it takes a lot of time.
Okay, sir. Next question, why steroids are used by some athletes?
So I will say the same question is vaccination or something. I'm not specialist of the vaccination or any kind of liquid injectable drug. So I'm sorry, so I don't have too much knowledge on the liquid injectable drugs.
Okay, sir. Sir, thank you so much. Thank you for giving us your valuable time. It was really great. Thank you, sir.
No problem. Thanks. It's my pleasure.
Okay. Now we are going to start our second session. Our next resource person of today's webinar is Dr. Ashwini Kumar. He is a native of Bihar and completed his BTech and MTech in biochemistry from Amit University and NIT Rourkela respectively. He was project assistant in a tissue engineering project at NIT Rourkela and then joined as an ad hoc lecturer at Department of Biotechnology, NIT Raipur. He taught immunology, drug designing and pharmaceutical technology, stem cells and healthcare and animal biotechnology. He joined the same department as full-time PhD candidate in 2014 and was awarded his PhD degree in January 2020. His broad area of research was developing mucoadhesive drug delivery system. He joined Department of Biochemical Engineering and Biotechnology at IIT Delhi as a research associate in March 2020. Now, I would like to request Dr. Ashwini Kumar to start with his presentation. Please, sir.
Very good evening to all. Am I audible?
Yes, sir. You are audible.
Okay. I would like to express my thanks to Department of Chemistry, Cooch Behar College, for giving me an opportunity to present a topic which is very relevant to the current pandemic situation. I'm going to share my slides. Is my slide visible?
Yes, sir. It's visible.
Okay, thanks. So today I'm going to present on immunity and vaccines, some general concepts and some relation to the current disease, COVID-19, as you all know. So this will be broadly my topics, basic concepts in immunity, basics of vaccines, and the different vaccine candidates we have for COVID-19. So, as you know, the immune system of our body is a complex network of organs, cells, and various proteins that are used against diseases, you can say. Mostly antigens, which are coming inside the body. I'll just start my laser pointer. Yeah. So, antigen is anything which is not recognized by the body at its own component, and it is coming from outside the body. So antigen, as you say, gen relates to generating. So antigen is something which generates the production of antibody. The immune system generally recognizes body components as self and non-self. And when the body recognizes something as non-self, it can result in different diseases and also in autoimmunity. So abnormalities of immune system can lead to allergic diseases, immunodeficiencies, and autoimmune disorders. So immune system is basically classified into three broad categories. One is innate and adaptive immunity. Second is active and passive immunity, and then cellular and humoral immunity. The innate immunity is something which is present from the birth, and it is general to all the antigens or to all the diseases. And innate component consists of macrophages, dendritic cells, natural killer cells, eosinophils, basophils, their skin, mucus, and complement system. These are general for, they attack every antigen generally. They are not specific to any particular thing. While the adaptive immune system consists of B cells and T cells, which are responsible for a specific generation of antibodies, which target a specific antigens. And these two cells are actually responsible for the immunological memory, which I'll cover later on in the next slides. Then active and passive immunity depends upon the antibodies. These antibodies are also called immunoglobulins. And the third classification is cellular and humoral immunity, where the cellular component consists of cells, which I have talked here, and the humoral component is consisting of antibodies and cytokines. Then the whole immune system is generated in the bone marrow. Bone marrow is the site where all the cells are produced. Then we have thymus. Thymus is a place where the T cells are matured. Then we have a spleen present on the left side of the abdominal cavity. It is a place where the blood-borne antigens are basically captured and processed. Then we have lymph nodes all around the body. These are the, you can say, the basic center where these immune cells functions. Then we have mucosa-associated lymphoid tissue. This is a component of immune system which is present around the mucosal tissue. This MALT consists of two broad divisions, gastric associated lymphoid tissue and nasal associated lymphoid tissue. I'll come to this in my later slides. So coming to innate and adaptive immunity, innate immunity is basic and general to all the antigens, and it takes minutes to hours to get into action, while adaptive immunity takes days to function. As I've said, innate immunity is present by birth. It's non-specific, is the first line of defense, and acts on antigen in the same manner. While adaptive immunity shows a specific action towards antigens or pathogens. Antigen and pathogen, you might know, like dust is an antigen, is not a pathogen, but bacteria is a pathogen. So every pathogen is an antigen, but every antigen is not a pathogen. Then among the immune cells, we have some specific cells which are known as antigen presenting cells, APCs. They actually process the antigens coming inside the body, and they present a particular part of that antigen to the cells that recognize that antigen as something foreign to the body and generate a required immune response against that antigen. So this innate and adaptive immunity, as I say, consists of T cells. The T lymphocytes generally secretes compounds which are known as cytokines, and these cytokines helps in function of B cells and other immune cells. The T cells are broadly classified into T helper cells and T cytotoxic cells. These are differentiated on the basis of presence of a particular protein on their cell membrane. The TH cells have CD4 protein on their surface, while TC cells have CD8 protein on their surface. And when these TC cells, they meet those antigen presenting cells which activate these TC cells, the T cytotoxic cells becomes cytotoxic T cells. You can just put this cytotoxics before the T. So TC cells become CTLs, and these cytotoxic T lymphocytes are basically a kind of phagocytes. They attack the disease cells and engulf the antigen coming inside the body. These are one kind of phagocytes. Then we have B lymphocytes, B cells. B cells basically are of two types, plasma cells and APC. B cells also function as APCs. The plasma cells, these are responsible for secretion of antibody. Then the B cells and T cells, they have a separate type known as memory cells, and these memory cells actually remembers the antigen, so that when in some later part of life the same antigen or the same pathogen enters the body, these memory cells are good enough to attack those antigens or pathogens. The T helper cells, which I discussed here, they actually activate the B cells to secrete the antibodies. So these antibodies are a part of adaptive, active immunity, and a passive kind of thing, active and passive both. Here you can see the CD4 T cell, the T helper cell, recognizes the part. This is the red part, the part of antigen presented by any antigen presenting cell, macrophage or dendritic cells, and this recognition and binding activates the TH cell. The mature T helper cells then again becomes either TH1 cell or TH2 cell. And this is a TC cell activation. When TC cell recognizes a part of antigen presented by another APC, it becomes CTLs. On my right side of the slide, you can see the basic structure of antibody. The tip of the antibody is the site which recognizes actually the antigen. This tip of antibody is known as paratop, and the part of the antigen that it recognizes and binds to it is called epitope. So this is a cartoon where ant asks his father, okay, why don't we get coronavirus? So the father says, because we have antibodies. So this is just a different spelling of the antibodies. I think my slides have full. It's not working. Hello.
Yes, sir.
Hello. I think there was some disturbance with the internet connection. My slides are frozen. It's not working.
I think it is internet issue, sir.
Might be. There's some problem with is it visible now?
Yes, sir. It is visible.
Okay,
now.
okay. Let me just go to the previous slide. Yeah. So sorry, I think there was some error. So yeah, I completed this slide. Now we have this, as I already say, the activation of T helper cell and T cytotoxic cells. So yeah. So these plasma B cells, when the immature B cells get activated with the help of T helper cells, these B cells become plasma cells and they secrete their specific antibodies for the particular antigen. This TC cell become cytotoxic and become phagocytic, and they attack the infected cells or the antigen which is not inside the cell. The antibodies can function in many ways. They can be neutralizing antibodies and they can behave as opsonin. When they function as neutralizing antibody, they can directly attack the antigen and destroy it. While when the antibody function as opsonin, it binds to the antigen so that the other phagocytic cells recognizes this complex of antigen antibody, and the phagocytic cells then attack this complex and destroys the antigen. So this is basically how T cell and B cell work together. This is a macrophage, a type of antigen presenting cell. When it engulfs the antigen, it degrades the antigen inside it, and present a part of antigen to the T helper cell. The T helper cell then becomes either Th1 cell or Th2 cell. The Th1 cell secretes cytokines so that many other cells can function well, and the Th2 cell they activate the immature B cell to the plasma cells for secreting the antibodies. Okay, then another type of immunity is active and passive immunity. The active immunity is when the body is exposed to pathogen, when we get a disease and we develop immunity against that. This concept is known as active immunity. For example, chickenpox. You can say once we have chickenpox, then we can be sure that for the next 15 to 20 years we won't get a second chickenpox. So this is a type of active immunity. While in passive immunity, we directly administer the antibodies to the body to fight an infection. Like you can see, there are different type of antibodies which are given as a drug, like rabies injection or tetanus injection. And in the current situation, the plasma therapy, which you might have heard, is also kind of passive immunity. So this is a basic diagram of how immunity works. You can see when the pathogen enters the body, these APCs become active. They present the antigen. The antigen is presented to the T cells. T cells again activates other cell and the B cell to produce antibodies and to function as a phagocytic cells. So this was the basic concept of immunity. So the whole concept of vaccines is based on immune system. Vaccine is a type of acquired or adaptive immunity and an active immunity, because we generally put up a pathogen, weakened pathogen inside the body, or a part of pathogen inside the body, so that the body recognizes that as a non-self and acts upon that. So when pathogen creates an infection, either directly or through some of its toxin, a disease condition is produced, then we have immune response against that, and we are cured or protected for that. The vaccine works like same thing. It is a weakened antigen which is presented to the body, injected to the body. The immune response is generated against that, and the protection develops. So this is a basic diagram of how vaccine works upon injection. So the component of the pathogen is generally recognized on the basis of PAMP and PRR. The PAMP is pathogen-associated molecular patterns, which are specific patterns or the molecules on the antigen surface, which are recognized by the immune cells. And PRR are the proteins present on the immune cells, which is pathogen-recognizing receptors. These PRR bind to the PAMP, and the required immune response is generated. So this antigen processing happens when it is inside the cell, and the antibodies are secreted upon activation, and antibodies can be opsonizing antibodies and the neutralizing antibodies both. The basic type of vaccines are, you might be knowing, inactivated vaccine, where the whole microorganism is destroyed by heat, chemicals, or radiation, so that the antigen or the pathogen cannot create the disease once it is inside the body. Then we have attenuated. In this process, the microorganism against which we need the immunity, it is not completely destroyed, completely killed, but the pathogenicity is weakened by the use of different chemicals. And then this weakened antigen or pathogen is used as the vaccine. Then we have toxoid. These are the toxins secreted by the antibodies, which are made a bit of a lower potency, so that they do not produce disease upon going inside the body. Then we have subunit vaccines. These are a particular part of the pathogen which are used as a antigen. This parts mostly are protein or the carbohydrate which are present in the pathogen. Then we have conjugate antibody. This when the subunit vaccine is subunit part is not strong enough to generate an immune response, it is attached to some kind of chemicals, and this complex is known as conjugate. Then we have heterotypic vaccines, where the pathogens that infect other animals but do not cause disease or cause mild disease in humans, like cowpox. the cowpox pathogen was used in early years to create immunity against the smallpox. Then we have monovalent vaccine, which generates only a single type of antibodies, very specific antibody. And then we have multivalent vaccines, where we have different, more than one antigen attached to the vaccine part. And these are the components which are used in vaccine: the active component, the antigenic part. We have stabilizers like magnesium sulfate. We have adjuvants that increase the immunogenicity of the vaccine. Then we have preservatives for preservation of the vaccines. We might have antibiotics in that, so that we can stop the growth of microorganisms, and there may be some trace compounds associated with that.
Sir, one second.
Yeah.
I request Chandrasekhar to please stop sharing your screen. Please stop sharing your screen.
Can I start?
No, sir, your slide is not visible.
Slide is not visible.
Sir, someone is sharing his
Okay.
screen. So-
Okay. Sure. I'll wait.
Yes, sir. You may continue now.
Yeah, thank you. So the production of vaccine requires the vaccine design itself. So the design of vaccine itself is a complex process that starts from etiology of the infectious disease. We have to know how the pathogen works, what kind of disease and what are the symptoms, what are the targets inside the body where the pathogen is attacking, this etiology and pathogens of infectious disease. And the identification of the protective immune responses, what kind of response body is generating against that pathogen. But most of the time, if the immune response is not strong enough, that's why we need the vaccine for preventing that disease. So this is a small snapshot from the Bharat Biotech companies. The vaccine development life cycle is quite long. As you have seen in the previous lecture, the exploration and R&D takes around 6 months to 1 year. And then we have preclinical stage that takes around 1 year. Then phase 1 trial, phase 2 trial, phase 3 trial, or similar to the drug development process. So if you combine all these timelines.
Sir, I interrupt you once more. Your slides are not visible properly.
Oh, okay.
Please, sir, check it once.
Sure. Is it visible?
No, sir.
Still not. Okay. Sorry. Yeah. Is it visible?
Yes, sir. Now it's visible.
Oh, yeah. Sorry. This technical things are creating some troubles also.
Yes.
So, yeah. This on the right side of the screen, you can see the vaccine development life cycle also takes around the same amount of time that you have seen in the previous lecture. It takes around in the R&D phase, it's around 6 months to 1 year. Then for preclinical testing, it's again around 1 year. Phase 1 trial, 2 and 3 trials, similar to the drug development process. And all around, it takes around 8 to 10 years of the time for a vaccine to come into the market. Till date, the fastest vaccine we have is for Ebola. That has taken around 5 years of time. But I don't know if that is working good or not. There are certain reports where the Ebola vaccine is not working great. So we have, don't go with the conventional vaccine types. I'll just jump directly into the subunit vaccine. As I've said, subunit vaccines are a specific part of the pathogen that creates the immunogenicity inside the body. The advantage with subunit vaccines is that we don't have reversal of pathogenicity. That means this type of vaccines don't create the disease inside the body. It just elicit the immune response. Like you might have heard, with oral polio vaccine, there have been cases where the child who got the vaccine developed polio. So this is what we don't want. Actually, we are protecting against the disease, but if the disease comes because of that vaccine, it is called reversal of pathogenicity. So with the subunit vaccines, there's no chance of reversal of pathogenicity. But the disadvantage is that most of the time the subunit vaccine have weak immunogenicity. So what is the solution for that? We use adjuvants. Adjuvants are any biological or non-biological component which are attached to the subunit part, and this whole complex is called conjugate vaccine. This subunit vaccine is either a polysaccharide or a protein, and the polysaccharide develops a T cell independent type of immunity, while the protein subunit creates the T cell dependent immunity. You can see in this picture, the adjuvant favors the presentation of the antibody. It helps the T cell for stimulation and further stimulation of B cells for proper antibody production, and it has different target actions also. It prolongs the time of presentation of the antigen to the APCs. So adjuvant is mostly selected, is very selective for a particular kind of antibody, for a particular vaccine, sorry. So in this chart, you can see the blue vaccines, they contain adjuvants, while the red vaccines, they don't have adjuvants. So this is a timeline starting from around 1800 when the smallpox vaccination was started. And this slide is only till 2010. So like the latest prostate cancer vaccine, which is currently in the trial by different companies, it doesn't have an adjuvant, while the influenza vaccine has got an adjuvant with that. The next is a conjugate vaccine. As I've told, it is a like subunit vaccine is attached to something which can increase the immunogenicity of that vaccine. So you can see when it is just a polysaccharide, a subunit, the dendritic cells captures it, present it to the B cell, but since it cannot elicit the TH cell response, the B cell eventually dies because of apoptosis, and there is no significant immune response. When a protein and a carbohydrate are co-formulated, they are not attached, they are just inside the formulation together. Then again, this kind of vaccination also does not elicit the TH response. The B cell recognizes the component, but eventually dies of apoptosis, and again, no significant immune response. But when these components are conjugated, there's a strong immune response where B cell recognizes the component, and the T cell is B cell engulfs the antigen, the T cell is again activated. T cell further reversely activates the B cell into plasma cells, which secretes the antibodies, and the memory cell for remembering the antigen for any further attack. Like you can see, this is a simple polysaccharide, then there is no memory, there is no activation of T cell. The polysaccharide, when it is attached to a carrier protein, conjugated to a protein, then we have proper activation of both the B cell and the T cells, and along with the memory effect. Then we have something called virus-like particles. In this figure, you can see this is a normal virus, and the inside is the genomic component of the virus, while the virus-like particles, they don't have the genomic component because these are synthesized so that the immune cells of the body recognizes these epitopes around the cells, but these virus don't have a genomic component, so that these virus do not create any disease inside the body once these particles are used as a vaccine. You can say this is a type of subunit vaccine. This viral-like particles, they elicit a very strong immune response, and they are primarily phagocytosed by dendritic cells, and these dendritic cells present the antigen associated with these VLPs using both MHC1 and MHC2. This protein MHC1 and MHC2, these are the proteins which are responsible for presenting the antigenic part to the TH and TC cells. We have certain VLP-based vaccines in different phases of trials, and these red names are the VLP-based vaccines which are already marketed. Then we have nucleic acid vaccine. These are, you can say, these are the latest in the team. You might have heard of Moderna company from this is a US based company that is testing its mRNA based vaccine. So in the nucleic acid vaccine, we have DNA based vaccine and the mRNA based vaccine. The DNA based vaccine, the component which is actually the DNA of the pathogen that encodes for the particular protein which is antigenic, it binds to the cell, the genome of the cells gets translated into protein, and those proteins are presented by the cells to APCs. Again, the same process continues. The B cell is activated and antibodies are secreted. But with the mRNAs, we don't have this. The mRNAs don't go inside the nucleus. They are translated only in the cytoplasm. So there is no chance of getting any kind of negative immune response because of the mRNA vaccines. So you can say the mRNA arsenal is much better as compared to the DNA arsenal. But mRNA vaccination technology is very complex, you can say. There's a lot of optimization which is required for mRNA to be a very suitable candidate. And these all are biological optimization process, like the each specific mRNA part or the sequence, they all need to be optimized. They all need to be, there might be some additions or deletions associated with that mRNA chain before it becomes a suitable candidate. So in short, these are coming to this pandemic situation. These are the vaccine candidates, types of vaccine, and below this chart you can see different companies which are into such kind of like Moderna and Inovio. They are into mRNA and DNA kind of vaccine. Then Codagenics and Indian Immunologicals, they are developing a live attenuated vaccine for COVID. The Sinovac and Sinopharm, these are Chinese company. They are developing an inactivated vaccine. Then Novavax in developing a subunit vaccine, and the current AstraZeneca and Johnson, they are developing a viral vaccine. This is a website where you can get the complete information of the vaccines under development. On the left, you can see the vaccines which are currently in phase 3 trial. And the right, again, different the vaccines which are in the last trial. You must have heard a few days back that the vaccine by AstraZeneca has been put on hold because of some unwanted adverse effect. But yesterday, it was again announced that the vaccine is now again in the race. There are certain vaccines in India also which are almost completely Indian made. One is Covaxin, which is made by Bharat Biotech. And this is the first vaccine which was announced by an Indian company. Then we have a vaccine from Zydus. This is Zycov-D. This is a DNA-based vaccine. The Covaxin by Bharat Biotech is an inactivated virus vaccine. Then we have Novavax, which has a tie-up with Serum Institute of India for licensing its vaccine for Indian population. Then the AstraZeneca vaccine is again has a tie-up with Serum Institute, and the trials are going in India also. Then Biological E, I think it is a Bangalore-based company. It has a tie with Johnson & Johnson for a COVID vaccine. This vaccine is a viral vaccine that uses adenovirus as the vector for transferring the antigenic component to the body. This is a very recent, it was I think September 11 news. The Covaxin from the Bharat Biotech has shown very positive response in the monkey, the preclinical trial in the monkeys. Then we have Indian Immunological company from India that has a tie with Griffith University for developing a COVID vaccine. Then this Minvax is a startup by this is a buddha from Indian Institute of Science, Bangalore. It is also developing a COVID vaccine. Then we have VLP candidates for this vaccine. This is a Medicago company, and the VLP candidate is currently in phase 1. Then from India, the Premas Biotech, it is based in Gurgaon. They are also introducing a VLP-based vaccine for this COVID. And this Premas Biotech, you can see it is developing the world's first triple antigen virus-like particle that presents 3 different type of antigens in the same component, so that we have much stronger immune response for the COVID. Then we have something from West Institute, USA. This is a biomaterial scaffold-based vaccine that slowly releases the antigenic component and that attracts the APCs very slowly, so that we have a elongated immune response. We don't need, we might not need regular booster dose or something like that. Then we have what is known as mucosal vaccine, which is targeted directly to the mucosa. As you all know, the COVID is basically a disease of respiratory tract. So the lungs is a mucosal tissue. So there are companies which are targeting mucosal vaccines, which can directly be administered to the nasal route or through the oral route. So why mucosal immunity is necessary? Because if the vaccine is injected through syringes, there are concerns that the mucosal immunity is not very strong. Since this COVID is a disease particularly targeting the different mucosal tissues, scientists feel that there need to be a mucosal immunity, a strong mucosal immunity against that. These are different examples where companies have developed nasal vaccines for developing mucosal immunity. In India also, Bharat Biotech is developing, along with University of Wisconsin, what is known as Coroflu as a mucosal vaccine. Then these are again examples of other mucosal vaccines that uses viral vectors. We have this company known as Vaxart that is developing a vaccine in the form of tablet. And currently, the vaccine is in preclinical trials, you can see from this chart. Then we have something called Ora Pro, which is a vaccine in the capsule form. It can be taken just as a simple capsule, and the antigen is processed inside the GIT, the gastrointestinal tract, which is a mucosal tissue, so that we can expect a strong mucosal immune response. This is being developed by the company, UK-based company called Stabilitech, and they claim that their vaccine do not require any cold storage, and it can be preserved at room temperature for around 2 years of time. Then we have amphiphile vaccines that exactly targets the lymph node. Lymph node are the most active center where the immunity, the whole process of immunity is generated. So there is a company in USA, Elisio Therapeutic, that is targeting its candidate vaccine to the lymph node directly by taking the advantage of human body protein albumin. The vaccine candidate actually binds to the albumin, and the albumin from the blood goes to the lymph node naturally. So when it goes to the lymph node, it carries along with it the antigenic component, which is very precisely targeted inside the lymph node, where a strong immunity can be generated. And the immune cells which are primed for this type of vaccine, they are very specific, and once they migrate to the different sites of body, they can generate very strong immunity against the particular antigen. So this is the snapshot from the very recent publication by the Elisio, where they have shown that their candidate vaccine has showed almost 25 times more higher antigenic T cell response, and the antibody titer was around more than 265 folds higher than the normal antigenic titer. This COVID vaccine is currently, I think they have filed the IND, the investigational new drug application with the FDA, and they will be soon starting the phase 1 trial. Then we have something called vaccine repurposing, where we don't formulate the vaccine from the scratch, but we see which existing vaccine can elicit required immune response against any particular antigen. So currently for this pandemic situation, we have a certain previous vaccines like BCG vaccine, which Serum Institute of India is currently trying to repurpose against COVID. Then there was a product from Cadila, which is known as Sepsivac. Cadila has also started trial at different hospitals in India. Then in Australia also, people have started working with this BCG vaccine against COVID. You can see this repurposing is actually in the trial. This is a snapshot from clinicaltrial.gov, where the clinical trials are actually registered. This was my last slide, and thank you for your kind attention, and I am open to the questions. Thank you.
Thank you, sir, for this informative presentation. It was really excellent and full of information. Thank you.
Thank you.
Now we start the question answer session. First question from Irene Ahmed. Are vaccines weakening our natural immune system? Is the immunotherapy safe as a cancer treatment?
Immunotherapy is something different than vaccination. Immunotherapy mostly consists of antibodies, monoclonal antibodies, which are given as a drug. But vaccine is not for treatment. Vaccine is for prevention. We try to prevent any disease from occurring when we are talking about vaccines. Immunotherapy is completely different thing.
Okay, sir. Next question. It is said that COVID-19 vaccine being developed by some countries is not effective for its co-types, but others claim that vaccine being developed by them is effective for all of its co-types. What is the difference in mechanism? How the same vaccine can be targeted for all of its co-types?
That depends. It's a multifactorial approach actually. The antigen which we are considering as a vaccine candidate, they can be different. Like you might have heard, we have N spike protein and S spike protein on the SARS-CoV-2 virus. So the choice of antigenic component that I've discussed in subunit vaccine type, that choice actually decides what kind of immunity will be generated, whether the immunity will be a long term or a short term. But that also depends on mutation. If the mutation is not very detrimental, that it is not changing the virus it's not changing the virus structure so that the vaccine is not acting. Unless that happens, the vaccine will almost work for every strain of the virus or the bacteria.
Okay, sir. Next question. Which type of vaccine is currently being developed against COVID-19?
As you can see from my slide, there are around 200 different types of vaccines are currently in different phases of development, and there are around 6 to 7 vaccines which are in phase 3 trial in India and across the world. There are Indian companies which have tie up with different companies abroad so that their vaccines are also easily available in India.
Okay, sir. Next question. Research paper said that before 20 years there is evidence of COVID-19. So why from before 20 years they did not prepare any vaccine against COVID-19?
See, research is a long process and ongoing process. It cannot start from scratch. If something happens, people were studying. If you have seen papers where people have studied coronavirus. So coronavirus is actually a class of virus. If you have heard of SARS pandemic in the China way back in 2002 and 3, that SARS virus is also a member of the coronavirus family. So the virologist or the microbiologist, they actually keep on studying various variants and various strains of the pathogens that might be pathogenic or they might not be pathogenic, but that research has to be a continuous process. So we cannot predict something if whether it's actually going to happen or not. But we can try. We can always try to develop drugs and vaccines against something which may come.
Okay, sir. Next question. Can we rely on vaccine prepared by Russia?
I think this is a disputed question. I'm not in a position to say anything on that.
Okay, sir.
But certainly, for any drug or vaccine, the thing is, if we are actually following the protocols which consist of all the preclinical and clinical trials, and we stuck to those protocols honestly, then ultimately the science has to prevail. It's not a race presently, or for any disease. There should not be any race. There should be science behind something. So I'm not saying that a vaccine cannot come in such a short such a short time. But if the vaccine has undergone all the required processes, then we can definitely rely on that vaccine.
Yes, sir. The last question. The last question is, as the vaccine for COVID-19 is being prepared even faster than Ebola virus vaccine, will it be effective enough? And what are the chances of relapse? Also, will it have side effects? If I'm
not wrong, Ebola was restricted to a particular region, and people were definitely trying to develop a vaccine as fast as they are trying for developing the corona vaccine. The Ebola vaccine took some 5 years, and scientists are claiming presently that some of the vaccines are currently in phase 3 trial. So if everything goes by science and by the protocol, we might expect a vaccine soon. As I said, it's not a race. Everything is science. So if everything is under protocol, we don't have any problem relying if a vaccine is effective or not.
Okay, sir. Thank you so much for giving us your valuable time. It was really great, and your presentation was really full of information. Thank you, sir.
Thank you so much. Thank you so much.
So now we are going to start our third session of today's webinar. Our next resource person of today's webinar is Dr. Goutam Biswas. Dr. Biswas is presently pursuing research towards synthesis of small molecules having therapeutic activity and nanomaterials. He obtained his PhD degree in 2006 in synthetic organic chemistry at Indian Institute of Chemical Biology. He has over 15 years of experience in different fields of chemistry like total synthesis methodology, organometallic, solid phase synthesis, and also in nanochemistry. From 2018, he joined as Assistant Professor of Department of Chemistry of Cooch Behar, Kanchanjunga Burma University. He has co-authored over 16 scientific publications and 4 patents. His research interests include carbohydrate chemistry, nanomaterials, total synthesis, and medicinal chemistry. Now, I request Dr. Goutam Biswas to start with his presentation. Please, sir.
Yeah, thank you for the kind introduction. I think I am audible, right?
Yes, sir.
Okay. And my slides are also visible, right?
Yes, sir. Your slides are also visible.
Okay. So thank you. Good evening to all. And first, I would like to thank our respected VC sir, who was here before, Professor Dev Kumar Mukhopadhyay, and then like to thank our registrar, Cooch Behar Kanchanjunga Burma University, Dr. Abdul Qadir Safeli, Principal of Cooch Behar College, Dr. Pankaj Devnath, and Dean of Science, CBPBU, Professor Prabir Kumar Haldar. I also like to thank Dr. Robindra De, HOD, Department of Chemistry, Cooch Behar College, and the teachers of this college for inviting me in this international webinar. Also, I'd like to thank all the speakers, teachers, and researchers and students who are present now in front of phone or laptop through Meet or YouTube today. So my topic is nanomaterials, a possible therapeutics in SARS coronavirus. So as we all know that since the beginning of 2020, coronavirus have taken a pandemic situation throughout the world, but today almost 3 crores of people have been affected by this virus, and around 9.5 lakhs of people died due to this infection all over the world. It has been almost around 10 months since the first coronavirus was detected in Wuhan, China. And obviously my previous speakers have already mentioned about some of the drugs and the vaccines which are in the race for this virus. But apart from that, as we all know, the nanoparticle is also an emerging field. So the nanomaterial chemist thought of using these nanomaterials in the fight for the therapeutics or as a delivery agent for these coronavirus drugs and the vaccines. So today I'll just give a very basic things about these nanomaterials and how it helps the researchers throughout the world or for having a good research about this SARS coronavirus. So today I divide my topic in 4 topics mainly. One is the introduction. Second one is the relationship between the virus and the nanomaterial. Third, I will just briefly discuss about some of the research work which is done in our lab in Cooch Behar University. And the last one is a conclusion. So first one is the introduction about the nanomaterials. So those who have much idea about the nanomaterials, I just give a very brief introduction about this nanomaterial. So if we consider a boy of 4-year-old is about 1 meter, then a nanomaterial is 10 to the power minus 9, so which is very small. Even it is smaller than the diameter of our hair. So this is a hair, this is a fly, so which is around 1 centimeter or in between 1 to 10 centimeter. Even it is smaller, the nanomaterials are smaller, nanoparticles are smaller than the red blood cell. But it is obviously bigger than the water molecules, which constitute almost the 3/4 of the Earth. So it is in between the molecular level and the microscopic or the macroscopic level. So these size actually makes a difference, because if it is a macroscopic, then it is very difficult to affect a very small particle. But if it is a very small, the molecular one, then obviously it can affect the virus. But since here you can see the virus size is around 10 to the power minus 7. So this is a hepatitis C virus. And obviously our coronavirus has also a size of 10 to the power minus 7. It is around 100 nanometer in size. I will show those pictures later in my slide. But yeah, so I think now everybody knows that what are the nanoparticles are. So nanoparticles are mostly the particles which are having their size in between 1 nanometer to 100 nanometer. So here it is. So as you can see, the nanoparticles are in between 1 nanometer to 100 nanometer. And generally the organic nanoparticles, the nanoparticles are of 2 types because of their origin, mostly. One is an organic nanoparticle, so which comprises of the polymeric nanosphere, polymeric nanocapsules, polymeric micelle, liposome, dendrimer, etc. And the inorganic nanoparticles, which are mostly composed of mesoporous silica nanoparticles, carbon nanotubes, silver nanoparticle, iron oxide nanoparticle, gold nanoparticle, and the quantum dots. So this is just a basic differences, means or the basic constitution of the nanoparticles. Apart from that, there are also many different examples of the nanoparticles, but this is just the basic one. And as here you can see here, the nanoparticles have different applications. It has applications in the environment, it has application in the industry, it has application in the food and agriculture, and even the nanoparticles have an application in the biomedical. So in the environment, now the people, in fact, we are also trying to develop the nanoparticle as an wastewater treatment in our lab. I will come those things later in my slides. Nanomaterials have also been used in the biodegradable of the polymer, in UV protection. These nanoparticles have also been used in industry as a reinforced plastic. Then this nanoparticle has also been used now as an industrial catalyst, functional nanocomposites, etc. These nanoparticles have also been used in the food and agriculture as a nutraceuticals, food packaging, reduced pesticides, nutrient delivery, improving texture, and these nanoparticles have also been used in biomedical also. So it has been vastly now researched all over the world for the cancer therapy. These nanoparticles or the quantum dots have been used as a imaging sensors also. These nanoparticles have been used in the protein aggregation, also in the drug delivery, and as an antibacterial agent. So here in our Cooch Behar lab, so we are mainly focused on those wastewater treatment, antibacterial, and the industrial catalyst. I will not go into the details of that one now. But yeah, so those red marked things we are now interested in our lab, in Cooch Behar lab. So I'll just show you a slide where we'll get an idea that how this nanotechnology was available in our real world. So as you can see here, a salt grain, which we see in everyday life in our kitchen, it is around 1 lakh nanometer in size, where the nanoparticle is only a 1 to 100 nanometer. So you can obviously think that how small the nanoparticle with respect to the salt in size. So these nanoparticles generally are used, or the silver nanoparticles are used as an antimicrobial agent. So it generally used as an antimicrobial agent as in order to damage the cell wall of the bacteria and thus inhibit the protein replication of the bacteria. So this is how the nanoparticle have been used. The nanoparticle have been used also as a sunscreen, means in the zinc oxide and the titanium oxide. So those protect the UVA ray and UVB ray. So when you buy a sunscreen from a store, you can see there is a number was written there, SPF 30, SPF 40, SPF 25. So those SPF, the full form of SPF is sun protection formula. So the sun protection formulas in most of the cases are nothing but those are the nanoparticles that they're mixed in a fixed ratio so that it can protect the sun rays to enter into the screen. So it can protect as a, we use those things in the as a sunscreen. Also the nanotechnology is used in the cloth. So in case of the jersey of the full of a player. So obviously there are sweat, so there are the fungal infections over there. So generally now the cloths are colored with the nanoparticles. So generally the silver coating was done for the antimicrobial prevention. Also the silica for the water repellation, titanium dioxide and zinc oxide. As you all see, the cricketers or the players, when they are out in the field in the sunlight, they used to apply the zinc oxide on their skins, in the bare skins. So there also nowadays the nanotechnology came into being, and the antimony doped tin oxide. So those are the applications in case of sports. Apart from that, in preparation of the sport equipment, like in tennis racket, now US Open was there. So I think tonight we'll be having a men's final. So there mostly most of the professional players are now using the tennis racket which are made of the carbon fibers, the carbon nanotubes. So because generally they are 100 times strong than that of a steel, means if the steel of the same diameter are made, then the nanofibers, the carbon nanotubes are 100 times much stronger than that of the steel. And it is also very light. So obviously it weighs like 1/6 of the steel, and also it is very much stiff. So these makes the most of the professional use the nano tubes, the carbon nanotubes as the string in the professional play. Also, the nano materials have been used as I mentioned before in the slides, that they are used as a quantum dots. So you can see that with the increase in the particle size, the color of the quantum dots change. So thus, we can use the nanotechnology as the sensors. This is the introduction about the nanoparticle. Then I will come about the relationship of the virus with the nanoparticle. So on the right hand side, you can see here, there are the nanoparticles like the magnetic nanoparticles, quantum dot, nanoemulsion, droplet, etc., etc. And on the left hand side, there are some virus structures which also have this nanoparticle size. And nowadays, the researchers all over the world are now combining both in order to develop some materials, nanomaterials, which will be helpful in killing some of the viruses, or can be used as a therapeutics as well as a carriers. So this is the relationship of the nanoparticles with the virus. So as we all know about now, I think everybody knows about the history of the coronavirus, human coronavirus. Though the animal coronavirus was first isolated in 1930s, I'll not go deep into those animal coronavirus, but I'll just give a very brief history about the human coronavirus. The first one was isolated or reported in 1965. So there, basically, it was reported as B814. In 1966, then another group in University of Chicago, they described the coronavirus. They work independently, and they described it as a 229E virus. In 1967, OC43 was also reported. That one was also coronavirus. In 1968, now this paper was published in 1966 and 1967. Then all the researchers who are working with the virus, then they see the images of the coronavirus, and they found that it has a crown-like spikes on the outside. So based on that, they named it as a corona because it's Latin, the corona means the crown. So they apply to the nature, and there in the nature paper, it was renamed this virus as a coronavirus. So that's how the coronavirus came into being in 1968. Then in 2002 and 3, the SARS coronavirus, which is the previous version of the recent one, was isolated in China, or reported in China. That time it doesn't take that much a pandemic situation, but yes, that time the similar kind of virus was reported. In 2004, this in Netherlands, the coronavirus was also isolated or reported from a child with pneumonia. In 2005, in Hong Kong, also the coronavirus was reported. In 2012, MERS coronavirus was reported in Saudi Arabia. So I think everybody knows about it. And in 2020, better to say 2019, the SARS coronavirus 2 was reported. So I think everybody knows that 2 it is because these coronavirus resembles almost like 85%, I think, if I am not wrong, as that of 2002-3 SARS coronavirus. So that's why it was termed as a SARS coronavirus 2 or COVID-19, because it was first isolated in or reported in December 2019. So this is just a brief history of the coronavirus. So those who want to know about the exact vision. Then come to obviously the family of the coronavirus. So how it belongs. As you can see here, the SARS coronavirus is in here, which is nothing but those are the beta coronavirus. But apart from the beta coronavirus, there are alpha coronavirus, gamma coronavirus, and delta coronavirus. So these 4 coronavirus are of the family Coronavirinae. And so these Coronavirinae and Torovirinae, so those are the 2 subfamily of the family Coronaviridae. And this Coronaviridae is also the order of this nidovirus. So this is how their family tree looks like. And our SARS coronavirus, so mostly you can see here, the human coronavirus are mostly either the alpha, beta, but there are some like Delta corona, HKU11 was also there. Yes, so this is the picture. So this picture on the right hand side is the tame image of the coronavirus of this COVID-19. As you can see here, the spikes are there. So I think everybody now knows how it looks like and all those things. So this is a schematic representation of the coronavirus. So this is a trimeric spike protein, so which basically first go and attaches to our body through the ACE2 enzyme. So in our lungs, and then there is an envelope protein was there, which is yellow colored. This is a envelope glycoprotein. This is lipid membrane. So this lipid membrane was also there. This is viral RNA and the nucleocapsid protein. So these, so when you wash your hand, like 20 minutes or 30 minutes, whatever, with a soap, then what we'll try to do is that we'll try to break this lipid membrane, because our soap also contains the lipid, and this is a lipid membrane. So when this cell ruptures, then this virus cell will die. So this is how we take the prevention from the coronavirus. In fact, when you use the alcohol, then also this virus also gets killed. So in that way, this is a mechanism basically how the coronavirus replicates within our body. So this is the coronavirus. This is a receptor which is present in our body, ACE2 receptor. And once this receptor takes this virus, it goes inside and this virus cell opens up. So this RNA, basically this RNA comes out, and here the replication takes place easily within our cell of this virus. And once the replication forms, then it forms again the virus, encapsulation takes place, and then this virus comes out of the cell by leaving the cell dead. So in this way, this virus replicates in our body. So this is just the mechanism of the virus. I am not going into much details about it, but yes. Okay, so we know the virus and the viral mechanism. Now comes to the nanoparticle. So I'll just give a very brief idea about the synthesis of the nanoparticle. So it's very simple. We can approach from both the sides. One is a top-down method and another one is a bottom-up method. So bottom-up method is the chemical reactions. When the chemical reactions takes place, then suppose when there's iron, when it was treated with oxygen, it forms the iron oxide. So one more atom, it combines with another, forms the FeO, or in that way. So with that, the size increases. So if we increase the size by the chemical reaction using the proper condition and the temperature, then it forms the aggregates, and when we control the aggregates, thus we forms the nanoparticle. There was another approach which is a top-down method. So top-down method, as you can see, there is a bulk metal, and if we crush it, it will form the powder. And when the powder was again crushed in a proper way by using the proper mechanical force, sometimes by sonication, then it also forms the nanoparticles. So one is a top-down, so that one is from the bulk metal to the smaller one, and that another one is a bottom-up, which is smaller one to a bigger one. So this is how mainly the nanoparticle synthesis can be divided. I'll just show you one nanoparticle synthesis. It's a graphene oxide coated silver nanoparticle. So graphene oxide, we generally synthesize the graphene oxide in the following way. We take the graphite flakes, add the sodium nitrate and the sulfuric acid to it, stir it for 0 to 5 degrees centigrade for 4 hours, and then the potassium permanganate was added to it, and then again stirred at 35 degrees centigrade and reflux, and then cooled, whereby the multi-layer or the monolayer graphite or graphene oxide stacks were found. And then the reduction of the excess of the KNO4 used here with the 2-mercaptoethanol was performed, and thus we get the graphene oxide powder. So this is the graphene oxide. Well, in some cases it is not nanoparticles. So what we used to do here, that we prepare the nanoparticles like from the silver nitrate. When we treat it with the ethylene glycol, pulse microwave irradiation, it forms the silver nanoparticles. And then since the graphene oxide was present in the solution, it is getting coated over the silver nanoparticles. So when we dry those nanoparticles in a vacuum oven, we will get the graphene silver nanoparticles as a powder. So this is just a very basic one of the examples, how the silver nanoparticles, graphene oxide silver nanoparticles was prepared. There are also various examples of the metal oxide or the organic nanoparticle preparation. I'll not go into the details of it, but yeah, it's very simple. Also, the nanoparticles are coated with different organic compounds, sometimes with organic compounds or with proteins, because in order to give it stability. Because when the nanoparticles, they live in the atmosphere for a long time, then they try to either oxidize or try to agglomerate to form a bigger size. So in order to prevent the agglomeration and the oxidation, we try to coat it, and just like paint. Like we used to paint the iron rod or iron sheet in order to prevent this oxidation. So similarly, the nanoparticles was coated with organic molecules or with the polymers in order to make it much more stable in the atmosphere. So these are the some of the stabilizing agents. Apart from that, there are innumerable number of organic stabilizing agents as well as natural products that are available in the market. So this one is the glutathione, this one is PVP, which is polyvinyl pyrrolidone, polydietyl dimethyl ammonium salt, PDDA. So the silver nanoparticles coating agent is the glutathione cap, silver sulfide nanocluster, which is GSH Ag2S, PVP Ag nanomaterials, silver coated GO AGNPs, of whose synthesis I have described before. Also, I have here there are also the PDDA, PVP, GOs, silver nano composite. So actually I am giving these names because now in one slide, next slide, I will show that these silver nanoparticles are used as a therapeutic agent for the coronavirus. It's not for the human coronavirus, but for the coronavirus. So here they are GSH Ag2S nanocomposite, or PDDA PVP GO silver nanocomposite. They are used against the porcine epidemic diarrhea virus, which are found in pig, and it has shown the antiviral mechanism. In fact, also these PVP silver nanowires or PVP silver colloids or PVP silver nanoparticles, they are used for TGEV, that is a transmissible gastroenteritis virus, which are also found in pig, and they have also shown the antiviral mechanism by disabling the cellular apoptosis. Also, this GO-coated silver nanoparticle, they are effective against the feline coronavirus, which are found in cat, and they have the antiviral mechanism through the inhibition of the viral entry. So obviously, these nanoparticles have known effect on the other animal coronavirus, but still now the silver nanoparticles have not been reported for the human coronavirus. So there is a scope over there. The gold nanoparticle also are there present there, which generally people use as a carrier or as a sensor. So as you can see here, the swine TGAV, which is found in pig, here the gold nanoparticle is used as a nanocarrier. Also the SARS-CoV, which is a human one, There also the gold nanoparticle are used as a vaccine adjuvant. I think vaccine adjuvant, they have an idea already because previous speaker, Dr. Ashwini Kumar, has already explained much more or much lucid way in that way. Also, the gold nanoparticle is used as an analytical sensor for the MERS-CoV virus, which are found in camel. Also, the gold nanoparticle have been used as an analytical sensor in human coronavirus. It is also used as an analytical sensor in case of PEDV. It is the gold nanoparticles also have an effect on the IBV, that is a bronchitis virus, immunobronchitis virus, which is found in chicken, and they are used as an analytical sensor as in both the cases. So there are applications of the gold nanoparticles as a sensors or as an carriers. Apart from that, where there are also evidence where the molybdenum, zirconium, molybdenum nanoseed are used in case of chicken as immunosensing. Zirconium quantum dots have been used for the nanosensors in case of the chickens. So they have been, so people have found their applications there. Ferritin-based nanoparticles, people have used as a vaccines over there in case of the MERS-CoV. And in case of the titanium dioxide nanoparticle, people have used the virus, it means the nanoparticle itself, as an antiviral agent in case of TGV and the PEDV. So those are the applications of the nanoparticles that have been reported till now against all the coronavirus, means both the human as well as the animal coronavirus. In some cases, the nanoparticles are used as an therapeutic agent, in some cases as a sensor, in some cases as a carrier. So this figure, I get it from ACS Nano, one journal, research paper, where their researchers have showed that the gold nanoparticle they used as a sensor in order to give the test for the coronavirus. So what they have tried to say in the paper is that after the sample collection, this coronavirus was given in this solution containing the gold nanoparticle, which was coated with some organic reagents. And then the coronavirus, when react with it, it precipitates. So once it precipitates, we can see it by the naked eye, so which means that it is positive. And if it does not precipitate, and after 10 minutes, then we can say that that person or that patient is the coronavirus negative. So in this way, they showed the applications of these gold nanoparticles in case of coronavirus patients. Apart from that, these nanoparticles can also be used as a coating for gloves. This is a coating for gloves. So in order to for the contamination, in order to prevent the contamination, it can be used as a self-cleaning surfaces also, like what I have recently heard that in Indian Railway, the toilets or the common places or the handles are coated, will be coated with the titanium dioxide nanoparticle, so which is to some extent antiviral and which prevents the coronavirus to be on that surface, to stay on that surface for a long time. And also these nanoparticles can be, people are thinking of using as a environmental sanitization or the sanifactions. It can be these nanoparticles, also people are now, researchers are now thinking of tagging it with the chloroquine or some other drugs to make it as a carrier. These nanoparticles are also used as a coating for goggles, mask, respirator, or PPE kit as an antiviral agent. Yeah. So those are the applications. So the nanoparticles have the applications, wide applications for these coronaviruses. But yeah, the peoples are now going for it in order to find a solution. So next I will go, I'll just give some 2-3 slides about the work which we are doing in our Cooch Behar University lab. So we recently published this paper in Nanomaterials, so which is titled that Green Synthesis of Characterization and Application of the Natural Product Coated Magnetite Nanoparticles for the Wastewater Treatment. So the first author is Chanchal, is my student, and the Shubhadeep is an alumni of the Cooch Behar College. So just give an idea. So since it's an iron oxide nanoparticle, so those nanoparticles have a magnetic character. So as you can see here, we place the magnet here, white color, and the nanoparticles get attracted toward this magnet. And also you can see, so this is by naked eye. Also we have done the squid experiment where the magnetic property was determined, and we found that here these graphs are very steep, so which proves that it has a super paramagnetic behavior. We have taken this same image from Korea, our collaborator, and there we can see that these nanoparticles have a globular-like shape. So these are the zoomed version, so which have a globular-like shape. So once we confirm that we have the nanoparticle, then we have done some biological studies. We have studies with the cancer cell, and we have found that it has no effect on the cancer cell. So this is one way good, because we try to use this nanoparticle for the water treatment, right? So after treating the wastewater, if we make it a clean water for drinking or something, then if the nanoparticle is in there, stays in there, if it is harmful, then it will not suit for the consumption. But luckily we found that it is not harmful to the cancer cell, but we didn't get the scope of checking it for the normal cell, but we are expecting that it will not be harmful for the normal cell also. We have checked it for the antibacterial properties from the wastewater. The wastewater we collected, and from there we isolate the bacteria present in that wastewater. We treat it with a nanoparticle, and we found that the bacterial concentration decreases on the treatment of the magnetic nanoparticle. So which is a good sign. That means that the bacteria present in the wastewater can be handled by these nanoparticles. Well, the wastewater contains the organic particles also, the organic compounds also, as well as the some inorganic pollutant like arsenic, cobalt, copper, etc. So in order to find those things, that whether our nanoparticles have any effect on those pollutants, we take a dye, because that will be much easier since it has a color. So we have found here, it's just for general, that this dye we take the methylene blue dye, and when we treat it with our nanoparticle, we have seen the blue color has been changed. We have done some experiment in our lab, and we have found that mainly this adsorption of the methylene blue dye on the magnetic nanoparticle takes place, and it follows the Friendly's isotherm model, and the order of adsorption is pseudo second order kinetic model. So I'll not go discuss all those things, just the graph is fine, I think. Also, we have checked the copper and the cobalt adsorption on this iron oxide nanoparticle, and we have found that it also gives a very good graph. And when we check the adsorption capacity with that of the reported nanoparticles, we have found, so those values are ours, right? So 463 and 543, so those are definitely much better than which are reported in case of copper as well as cobalt also. That means it was 513, 501, but where the reported one are 43.3 and 135.5, etc. Also, we get a good results for the methylene blue dye adsorption also. The reported nanoparticles are in hundreds, while ours is in 466. So obviously this makes us happy, and we can publish our paper in a very good journal. So this is about our work which we have done here in our lab. In last, I can say the nanoparticles are biodegradable and target specific, so they can be successfully employed in the production of the nanocapsules for the delivery of the fungicides or the antiviral agents or the drugs or the vaccines. In the drug means the delivery of those things. Nanoparticle will obviously revolutionize the agriculture, including the crop protection in the near future. And I hope all the nanoparticles researchers also hope that over the next 2 decades, green and the golden revolution will be accelerated by the means of nanotechnology. With this, so this is my small group over here. So this is Arindom Shah, who is also a teacher in the Cooch Behar College. This is Shubhadeep Chen. He is an alumni in Cooch Behar College. This is Chanchal. He is also a teacher in Alipurduar. Not in Alipurduar College, but in college in Alipurduar. So these 2 students are from Pune. They are synthesizing some compound. This is Raju and this is Sandeep and this is mine. So at last, I would like to say thank you all and stay safe. Thank you.
Thank you very much, sir, for this informative presentation. And we have got some questions during the session in our chat box.
Okay.
So first question. The first question is, usually receptors don't accept or allow foreign particles or unknown entity to our body. But why or how coronavirus was allowed by receptors or entered inside our body system?
Yeah, actually our body has some immune system which prevents the foreign particles to allow inside. But actually that's the reason why the coronavirus is being so pandemic, because the spike protein matches perfectly with our receptors. That one is the H2 inhibitor. So we can say it's our bad luck that these spike proteins of the coronavirus matches perfectly or sits perfectly with H2 inhibitors of our body, and that's why they sits there. But as soon as they sits in the receptors, they are not entering into the cell. They take 3-4 days to enter into the cell and for the replication. But yes, means so that's why the people who first comes in contact with the coronavirus patient get the symptoms maybe after 3, 4, 5 or generally after 7 days. So that's the reason why.
Okay, sir. Second question, whether gold and titanium nanoparticle antivirals will be cost effective to bring it for common people use?
Gold and titanium antiviral. Okay. So since it is a nanoparticle and people are now looking for the possible therapeutic treatment, now if it is found that this gold and the titanium oxide nanoparticle are much more effective than the compound itself, then obviously I think people will go for it, because in all the people will require this antiviral in a very small doses. Because when we take the pill, it goes into the stomach and then it works. But in case of the nanoparticle, if we can formulate an oral inhalation or something like that, then we require a very small amount so that it can go directly into the nasal way, into the lungs. And so that's why we require very small doses, and hence it will reduce the effective cost of the treatment, maybe.
Okay, sir. Last question. Application of gold nanoparticles for detecting COVID-19 is very interesting. Is there any commercially available nanoparticle-based drug for any disease or in process of preparation?
Well, it's a good question, but till now, what I have found, it's not in the commercialization, but there are evidences where some of the nanomedicines are in the phase trials. So obviously, since those are, as my previous lecture means the presenters have also speakers have also mentioned in their slides that it generally takes like 10 to 12 years at least to get those things into the market. And so I expect the nanoparticles which are in the phase trials will be in the market maybe in next 3-4 years or 4-5 years, something like that.
Okay, sir. Thank you very much for giving us your valuable time. Stay safe. Thank you, sir.
Thank you.
So now we are going to start our last session, and during this session we will share the feedback form in the chat box. So please stay with us. Our next resource person of today's webinar is Dr. Arindom Chatterjee. Dr. Chatterjee is from West Bengal. He did his BSc with Chemistry Honours from Ramakrishna Mission Vidya Mandir, Belur Moth in 2009 and MSc in Organic Chemistry from IIT Madras. He joined University of Connecticut, USA in 2012 as a PhD graduate student. His PhD research was mainly focused on the synthesis of site-specific DNA adducts of aromatic amines and nitroaromatic carcinogens and study of their replication properties. While doing PhD, he got an opportunity as an intern in a pharmaceutical company where his work mostly involved in various synthetic methodologies development. After finishing PhD on December 2018, he joined Wake Life Sciences, a biotechnology company in USA, as an oligonucleotide process scientist. So now I would like to request Dr. Arindom Chatterjee to start with his presentation. Please, sir.
Okay, so can you see my slide?
Yes, sir. Your slides.
Slides are up. Okay. Thank you very much. And thank you, Samanti, for your kind introduction. And I would like to take the opportunity to thank Dr. Snehasis Bhakto as well, because he invited me first actually about this seminar, and obviously Dr. Robindra Dey, the HOD of Cooch Behar College, for inviting me and having a slot for this presentation. So today I'm going to talk about the gene silencing in drug discovery, which is kind of a very exciting field, which is developing right now. Right now means since almost like last 10 years, trying to tackle some really genetic diseases. So I would like to present in these outlines. So let me get the laser pointer. Okay, so these are the outlines for my talks. I will touch upon some of the basic concepts of molecular biology, which is required to understand the process, and then the mode of actions of these oligonucleotide drugs, challenge associated with this drug design and synthesis, and then obviously some other late stages oligo development drug problems and how we actually overcome those things. And at the end, where the field stands right now, like what are the current progresses I've made in the last 10-15 years. And another disclaimer, whatever I'm presenting today, basically none of them are actually my work. So these are some of the journals which I cited here, and they are like excellent reviews. So if someone is interested, you can go back and check and read all these papers. So that will give you like a tons of informations which are really exciting. And I hope maybe I think at the end of the seminar, we are going to submit the PDF format. So if you want, you can check these links. So before I start, so the first thing, before we actually talk about gene silencing, the first thing which we need to understand what is gene and what it actually does. So gene is nothing but a DNA sequence, and what it does, it basically preserves your physical and like basic physical and functional unit of heredity. So it preserves all this information and pass from one generation to another. And in the first cartoon here, you can see the basic idea of the concept called central dogma, and what it actually explains. It explains how from a DNA, a protein is synthesized. So you have a DNA inside a cell's nucleus, and inside the nucleus you have the chromosome. So basically inside the chromosome you have a double helix of DNA. How it actually looks, it looks like this. Though it's a spiral like a double helix ladder, but in the form of chemistry, like if you look at the chemical structure of the DNA, it will look like this. And these are the actually three different parts of any nucleic acid sequence. This pentamer, like this furanose ring, is called sugar moiety. You have a phosphate backbone and you have different bases in this nucleic acid. And the way the chemists name it, it's actually a strand, like this is a double strand, the red and the blue. And this is, say, for example, one single strand, and it runs from like a 3' end to 5' end. And here you see these are the bases, and these are the names, like from 3' end, adenine, cytosine, guanine, and then tyrosine. And what it does inside the nucleus, it actually gives a transcript to synthesize the RNA. And once you have the RNA, what it does, it actually translates into protein. So you will have a RNA from the DNA, and then that actually forms a protein. Why this concept is really important? Because a lot of diseases which we are nowadays see is basically the root cause is different types of faulty protein synthesis. So if you have a faulty protein synthesis in your body, then it actually turns out really fatal. Like, say, for example, cancer. When cancer happens, means it's basically unregulated growth. You can't control the cell division. And the reason is a fault between DNA and RNA. When it actually transcribes, it gives you wrong information. In this slide, I will try to show you another like effort of humans, which is like humongous, and you will see the human genome, and the project took 13 years to finish. What it actually does, what they actually did actually, so it's literally sequence the entire human genome. Entire human genome means you will have 23 pair of chromosomes, 22 we call autosomes, and 1 pair of sex chromosomes, which determines our sex. Now, total 23 pair of chromosomes. If you sequence all 23 pair of chromosomes, you will get more than 3 billion base pairs. Now, you see it took 13 years to get all the information from the human chromosome. And the beauty of this project, once it was finished, I mean, it's still ongoing because there are still certain gaps which people have not actually figured out yet, but they're still working on it. You will see the different types of proteins which are synthesized in human body, and the location of those chromosomes, like the DNA sequence for this particular protein, it presents in chromosome number 13, and it codes in this under this the name of that BRCA2 gene. So gene is nothing but a DNA sequence which helps to produce certain proteins. And another thing which I want to mention here in the last 4 columns, you will see the length of the particular gene. So this BRCA2 gene is length is 83,000 base pairs, which basically consists of both your exon and intron, which means in a gene there are 2 segments which actually produce the protein and which is basically non-protein sequence, so which does not actually take part in your protein synthesis. So that part is called intron, and which is actually giving you the information for the protein production is called exon. So this particular gene is having 27 exon, and this 27 exon is actually producing that particular protein. Same for this, the last line, the dystrophin gene, which is the largest gene in human body, and it consists of 79 exon, means the 79 patches of a DNA is required to produce the right gene, the right protein, which actually a proper functional protein. So once you have a DNA, it forms the RNA and it forms the protein. So that's the basic concept. So here you have a DNA, you have RNA. So the only difference between a DNA or RNA is the structural difference. In the DNA, here you don't have a particular group. In the RNA, you have a particular group. And there is one base difference. In DNA, you have T. And the RNA you have U. That's the entire thing. And once you have RNA, the particular process called translation, it gives you certain protein, which is nothing but a polypeptide chain of different amino acid. Okay, so the next concept comes out is like the antisense oligonucleotide and the RNA interference, what it actually does. So antisense oligonucleotide, if you look at this cartoon at the bottom portion, so this is the human chromosome, and if you unwind it, you will see the gene. So this is the double helix of the DNA. And here inside the DNA, you will see one is called sense strand, one is called antisense strand. The sense strand is actually replicates to give you the same DNA sequence, whereas this antisense strands is actually helpful to generate an RNA transcript. Why is called transcript? It's having the word script, means I'm giving you some information and then you produce something. So you will get the RNA transcript, and once the RNA transcript is ready, there is one particular cell organelle called ribosome that actually produces a protein. So the first report came out in 1978, where these two people, like two scientists, Jeminick and Stephenson, they actually showed the first RNA dependent protein, a disruption, like it actually helps to stop the replication of that particular Rose sarcoma virus. And since then, people are actually started more progress, like how far this field can go. And then around 1998, the breakthrough publication actually came out. And based on that, what they did, these two people, Andrew Fire and Craig Mello, so here you see they actually, instead of a single-stranded RNA or DNA sequence, they put a double-stranded RNA here in the third cartoon. So when they put a single-stranded, it never happened. Nothing happened in this particular worm named C. elegans. Here you can see it's a regular normal worm. Even if you put an antisense, just a single strand, it never done anything. But once you put a double-stranded RNA, you form something like there is a difference in muscle protein. So which actually now, like a regular to it, started twitching. So this is the breakthrough, what happened like 1998. And based on this discovery, Andrew Fire and Craig Mello got the Nobel Prize for the discovery of RNA interference in gene silencing by double-stranded RNA. So which was huge in 2006. And since that technology came out, a lot of biotech companies are started like mining it more, that how far it can go. So this slide actually tells you the mode of actions of different oligonucleotide drugs. So we call it oligonucleotide. Basically, all these bases, when you combine, we call it like an oligonucleotide, like ATGC, like a sequence. So mostly this technology target the messenger RNA, which actually comes from your DNA after transcription. And under this antisense oligonucleotide, you have two different versions. One is a regular ASO and this other is called siRNA or small interfering RNA, which is a double helix of RNA. And you can see here different types of action. So something is called RNA interference, and here you will see the sequence length, how many bases you need to actually achieve that particular mode of action. Some of them requires 21 nucleotide, some of them are more than 21, some of them are like less than 20. It depends on the particular process you want to actually mine more. So here, another difference of, if you look at this particular panel, so different types of...
Excuse me, sir, 1 minute.
Yes.
Abdul Farooq, please stop to sharing your screen. Abdul Farooq, please stop sharing your screen. Okay, sir, you may start now.
Okay, so as the mode of actions of different nucleotide drugs. So if you use just...
You have to present your screen again.
Okay, sorry for that. Stop sharing. Just a minute. So now you can, right? Can you see?
Yes, sir, now it's okay. So now it's okay.
Okay, thank you. So here you will see different type of potential nucleic acid and the mode of different type of action. So you will see RNase H-dependent antisense oligonucleotides, something's called splicing modulators, sometimes called steric blockers. So I will go one by one about all this concept in a minute. So the first thing comes out is the antisense oligonucleotide. So look at this cartoon. It basically represents a cell. So here inside you have the nucleus and you have the cytoplasm of any eukaryotic cells. So when the transcription happens, which is basically forming DNA to RNA, you will get a single strand of pre-microRNA. So this yellow and green thing's actually your exon or intron. So the yellow thing is basically intron and the green thing is called exon. Why is it? Because you need that entire exon to produce certain protein. So think about a moment, like if you now produce a faulty gene, which means you're going to produce a faulty protein, and that can be fatal. So you need to stop that one. How you actually arrest a faulty gene here? So if you insert an antisense oligonucleotide, it actually works in two different ways. Once from this transcription method, like post-transcriptional modification, which happens to produce a mature RNA, it can actually stop at this level. So here you can see it binds in 3 different spots, not all at a time or maybe at a time, and 3 different types of mechanism works on particular areas. So if you put that here, so it basically forms it actually inhibits the 5-cap formation. If you block here, it will literally stop the RNA splicing. Splicing means you basically get rid of this yellow part and then you will get all the green part together. And same for RNase H activation, which means if you bind it here with the RNA, there are certain nucleases called ribonucleases H that triggers a degradation of this mRNA. So you will not going to achieve this particular thing. Now, if you achieve this particular thing and it's still a faulty, if it's still a faulty, then it will still produce a faulty protein. But if it's not, then you're all good. You will get a good protein. But if you're still having some faulty regions, like there are some mutations or something happened, then again, you can actually stop it at this center. So you will get a antisense oligonucleotide. Hybridization means basically binding with DNA and RNA. So antisense oligonucleotide is mostly based on the DNA. So it's a DNA sequence which binds with RNA. And once the DNA binds with RNA, this particular protein RNase H actually finds this location and cleaves this mRNA. So you are not going to get the false protein. Or even if it's RNase H is not activating, then once you have the duplex of this hybrid DNA RNA thing, when the ribosome actually starts making protein from this mRNA, it cannot actually sense this particular region here. So you are not going to get a protein out of it as well. So basically you stop the translation. So you can stop producing a faulty protein. So another concept is the double-stranded helix, which basically your RNAi or RNA interference using small interfering RNA. So how it actually works in a double-stranded RNA, which produced from your DNA. So though it's a cartoon, like looks like a ladder, but in real term, it actually forms different types of loops, like a hairpin loops. And it's a very long sequence of more than like 70 nucleotide. So when it forms like this particular hairpin loop, there is a certain protein called dicer. It actually detects that loop and it dies, means basically it cuts into smaller fragments of this double helix. So you will still get a siRNA, but it's now a duplex, short in length, like around 20 MER in length. So once you have a double helix of a very short sequence of 20 MER, there are certain proteins inside the cell called argonate2, dicer, and then TRBP. These are all in cartoons. It actually sends this double helix and then bind with that double helix. Once it binds with this double helix, it forms a complex called RISC or RISC, and the full name is RNA-induced silencing complex. Once that formation of that silencing complex happens, out of these two strands, one is called guide strand. Say, for example, the bottom strand is guide and the top one is passenger strand. It basically cleaves the passenger strand. So this upper strand goes away and it's now only binded with this double, like the bottom guide strand. Now the function of this guide strand is it now looks for a mRNA sequence which is complementary of these bases, like those small sticks. These are basically your AUGC. So once it finds a gene which is basically complementary of this particular guide strand, it binds with that guide strands, and this entire complex RISC, it actually sends this double helix. So once it binds with this double helix, it forms a complex called RNA-induced silencing complex. Once that formation of that silencing complex happens, out of these two strands, one is called guide strand, the top one is passenger strand. So this entire complex cleaves that mRNA. So if you have a faulty gene, you can still stop it in using this particular concept. So these are actually the backstory from where the field actually started, that antisense and sense your siRNA. Now, why it is important? Because it is important in certain disease areas. Like mostly when you take a pill, it's a small molecule drug. So what it does, it actually somehow disrupts your protein production, maybe using like terminating certain enzymes or making some mess with that particular enzyme so it cannot produce that particular protein. But there are certain areas where you still require certain medicine production or development of medicine which is still not available. So most of them are like genetic diseases or neurodegenerative diseases. Like genetic diseases are basically causing from your faulty gene. It can happen in different tissues, like even in eyes or in your liver or in muscle or central nervous system, like your spinal cord or brain. And you can see some of the names like Parkinson's or Huntington disease or ALS. These are neurodegenerative disease, which basically disrupts your motor or neuron function. So you literally kind of lose your control. So you cannot actually perform your day-to-day job, and at the end it becomes fatal. So having these diseases are really fatal, and there are no current cure of these diseases. So there are certain drugs available which basically slow the progress of this disease, but they cannot stop this disease. So nucleic acid therapeutics somehow having that promise that if they can develop, so they can actually tackle these diseases. And another part which in all the previous talks are having your COVID related, so it can still somehow block some viral infection or RNA. So this technology can also be helpful developing some of the vaccines which actually stops your viral RNA replication. So you can still get benefit out of this concept. So this slide actually tells you about a process called exon skipping. As I told you in the previous slide, there is a disease called Duchenne muscular dystrophy, which is actually a faulty kind of your protein production for your muscle. So it happens mostly young boys, like 3, 4, 5 years ages of boys. And why is it fatal? Because it cannot produce this particular protein called dystrophin. And the first slide, if you remember, I showed you a total 79 exon is required to produce this particular protein. So this is a cartoon presenting you all 79 exons. So you need this entire sequence to get a protein. But if you have a fault in any one of these particular position, say for example at 52, and if you look at this cartoon, when it actually joins or when ribosome produces protein after having this transcript, and if you have a faulty protein, faulty gene here or a mutation or something happened, then it cannot actually, and if you omit this one, this straight line cannot actually joins with this particular arrow-shaped thing. I mean, it's very cartoon kind of representation, but it's actually in terms of amino acid and in ribosome. So if you have an issue here, it cannot actually join with 51 and 53. So what it does, so you put a siRNA or antisense oligonucleotide to basically eliminate that particular sequence at 53. So you are deliberately skipping that 53 sequence so that 51 can still match with 54. This straight line is now matching and you can get a sequence like a straight line. So what it does, it actually still can produce a truncated protein. You may not get the entire like healthy protein, but you still can put some like a backup, some sense of protein production is still happening. So you can literally lengthen the longevity of that particular patient. So these are certain things which is happening in US, and there are certain companies which are still working really hard to develop this type of medicine. So at the end of the presentation, I will show you all the slides, like all the drugs which is approved till to date by FDA, and some of them are addresses this type of neurodegenerative or your genetic disorders. Now, have that particular concept. Another hurdle is how you actually design a nucleic acid drug. If you just say, for example, look at a sequence of mRNA which you want to target, and then you design that complementary sequence and you just inject it into the human body, it won't survive. It will The first problem is it cannot penetrate your cell when there is no modification, and it readily degrades by your endonucleases or exonucleases. So within a minute, it destroys. So this naked form of nucleic acid, it cannot help anything. So when this problem arises, so scientists actually look at the structure of the drug. So if you pay attention here at the red box, as I told before, the nucleic acid is actually made of different portions, so sugar, phosphate, and the base. The first thing they did, they changed that phosphate backbone, which is a charged backbone with oxygen. They put a sulfur on it to see how that affects that characteristic of that particular drug. When they did that one, it's good because it cannot degrades pretty soon, so it increases the drug's half-life. So when you inject the drug inside the body, it has to survive inside the body to do its job. And it's more effective to activate RNase H activation when you put a DNA sequence, so which is very good news. But still the problems are when you put just one particular modification, it's still very non-specific. It binds with different proteins. It's still having some toxicity effect. The binding is not that high, and obviously cell cannot uptake that particular drug very easily. So with this problem, then again you go back and then check what other things you can do. So it comes out after that is like second and third generation of the drugs, which is now all different types of modifications. Instead of having just a phosphate backbone, you started making different types of modifications in sugar, in the bases. So these, all the images are actually showing you different types of modifications, and what it does when you put different groups here in R, it changes your shape or the conformation we call of this particular ring. So all these conformational changes actually changes the property, so you will achieve certain good results. So maybe more better binding, more better efficacy. So even if you have this particular monomer, if I go back to the previous slide, so you have a monomer, if you cut it here, and then you need to make a sequence, which is the next hurdle, how you actually produce a oligonucleotide sequence. So we don't use it like in a regular day-to-day organic chemistry lab, like where you actually run a reaction in round bottom flask or in small vials. So this is the instrument which actually produce your DNA or RNA sequences. So these bottles are containing all those individual bases, and then this is a column where the entire reaction happens. So you will get the entire oligo sequence out of this column. So there are certain very technical parts which I am not going to talk about, but if someone is interested, we can discuss it later. So and the entire process is automated. Automated means you write a code in a computer and then you give the information to the machine, and that actually gives you the entire sequence. And there are different stages of this particular sequence, and you need all the thing is the monomer and certain reagents to achieve that sequence. Once you have achieve a particular sequence which you want to use as a drug and with all the modifications, so there are certain benefits. So you now increase the nuclear stability, binding affinity, and there are you can actually overcome certain immune stimulation. Immune stimulation means when you administer a foreign particle in your body, there are certain checkpoints in your cell which actually triggers it as like a stimulation, like your fever or something like that. And then you obviously achieve some very specificity of gene knockdown, but there is a problem with cellular uptake. How you actually deliver a particular drug in a very specific target. So there actually comes the role of formulation. So there are different ways you can formulate a drug. So when you actually take a pill, it's having the API or called active pharmaceutical ingredients, which is this thing, but you need to formulate that API in certain way so that a person can take the capsule or the tablet. So how you actually make that formulation for this particular genetic or nucleic acid drug, there are like the nanoparticle related formulation called lipid nanoparticle, or you can make a conjugate of this particular drug. So conjugate means is basically I will come back here. So conjugate is basically you are like attaching a tag with this particular nucleic acid sequence, so there are certain receptors in our body which recognizes this tag and then absorb this drug. So depending on your target, where you actually want to deliver this drug, you actually add that particular tag, and we call conjugate formation. So up to this point, you have a very nice sequence with all different types of modifications, and you achieve very good result. But still, because of certain other issues, next thing what appeared or scientists have discovered is the chiral nucleic acid sequence. So the first line here, it actually tells you a PS sequence, which means all the phosphodiester is actually now replaced with the PS, phosphorothioate sequences. Now, once you put a phosphorothioate, if you look back here, this particular area, it now forms a chiral center. Now, if your synthesis is not very much like stereopure, you're basically generating a huge number of isomers. So if you have a 20-mer sequence here and you have all these chiral centers which are not regulated, so you are basically getting total number of molecules is like 524,000. So every single time you dose this particular compound, you are basically dosing an ensemble of all this number of different proportions of this particular sequence. So where I work is Web Life Sciences. They have developed stereopure nucleotides. So now they can actually control each particular phosphorothioate centers depending on your stereochemistry. Either it can be R or S. This is a very technical term for organic chemistry. So those who are having that background, you know what I'm talking. So it's basically the different types of conformations of this particular center. And when you have a different stereochemistry at this particular different region, the property of this particular sequence changes, like the binding affinity, the efficacy, the toxicity, all these things. And as this antisense or siRNAs actually requires some protein, like if you remember argonaute 2, dicer, all these things. So protein is basically nothing but a polypeptide chain. Like think about your hand. So certain things is easily you can grip, certain things you cannot because the shape of that particular object. Same for a drug. If you give the right conformation, it's easy for that particular enzyme to actually handle this sequence and give you the better results. So this is the concept all behind the synthesis of your chiral nucleic acid drug development. And this particular sequence is nothing but the older version of your oligonucleotide drug development, like 2 slides back, what I showed you. But the only thing is you manipulate this particular area of that. So you're basically getting a stereopure oligonucleotide instead of a regular oligonucleotide. This particular thing, this slide actually tells you about, again, like the very specific delivery of these nucleic acid drugs. So depending on, as I told before, depending on what you actually tag with this nucleic acid, different types of ligands, it basically attaches with this particular cell. So if you want to achieve a very good result in GalNAc using these GalNAc conjugated oligos. Now the GalNAc receptors are actually expressed in liver cells or hepatocytes. So the moment you tag a GalNAc with your sequence, it actually easily, and you inject that one, it actually goes and bind with the liver cells. So you can actually tag and then achieve very different types of efficacy with these different types of siRNA. And Alnylam Pharmaceuticals, which is a biotech company in Cambridge, Massachusetts, they actually first developed this GalNAc thing. So, and this slide was actually taken from their website. And this slide is basically a more detailed cartoon of how that GalNAc thing works. So look at this sequence. Is either antisense oligo, which is a single strand, or siRNA, which is a double strand, and you put a linker, you put a GalNAc tag. And this is the surface of a hepatocyte or your liver cell. The once it reaches there are certain mechanisms which helps to internalize your drug, which goes inside the hepatic cell. Then depending on the pH of that cell, there are some changes in the conformation. So what it does, it clips that tag in the linker here and it releases that double-stranded or single-stranded RNA or DNA. And then with all the old explain mechanism like the risk, dicer, and all these things. It helps to silence a particular gene. And this receptor goes back and then it again actually takes the drug, and then this cycle actually repeats. So depending on the receptors and the cells, so you basically tag that particular conjugate. So far we have synthesized different stereo-pure or stereo-regular oligonucleotides with different types of conjugates. But even you make a sequence, there are a lot of work which actually goes behind before actually a drug comes out of a company. So first thing, if you remember, like the very first seminar presented by Anamitra, there are certain stages of drug development. First thing is your target identification, like which mRNA you want to target. And all these informations are coming out like using computers or like bioinformatics. This is an entire field which works on target identifications, or where it can actually bind, your drug can actually bind, how it binds, so all details. So they actually do using computer simulations. So you get the data from the bioinformatics team, you finalize, okay, we need to target this particular gene, and we see if we can knock it down. And then you screen like thousands of sequences which you think can be a good candidate for a particular disease. So another work is which requires like huge effort is from lead identification from thousands of molecules. So you get the hit results using again bioinformatics, and then once you have those initial result, like you narrow down from 1,000 molecules to 10 molecules. Now you want to test that 10 molecules on that particular target. And then before it goes to a clinic, there is a study called preclinical study, where it actually studies that pharmacokinetics or pharmacodynamics of that particular drug, the toxicity issue associated with this drug, how much dose you want to insert into a living organism. It can be a mouse model, it can be a regular assay, it can be your non-human primates, means monkeys or chimpanzees, or it can be human. So all this work you need to do before it actually goes for a clinical trial. And once you have all the data, you now finalize a candidate. Okay, we will go forward with particular sequence. Then you have to make sure you have a robust process to commercialize. Like even if you get a result of a particular sequence, you have to make sure every time you produce this molecule into your lab, it maintains certain quality. So quality control and quality assurance of a particular drug is also really important before it goes into market. And then once you have all these things ready, then you start your clinical trial, where you see to the actual human it works or not. And if you get a very good result, and there are certain phases of human trials. You can see clinical trials like phase 1, phase 2, phase 2B, phase 3, all these things. And if you succeed in these clinical trials, then you file if you file for the approval to the FDA, which is the regulatory body for United States, which is the Food and Drug Administration. For Europe, it's called EMA, I think European Medicine or European Medicinal Agency or Medical Agency. And then with all this data, you have to submit to them. And if they're happy or if they're satisfied with your data, then only you will get the approval. So this is the entire story behind the drug development. And this chart actually tells you the entire journey starting from 1978 to till today, how the academic research has helped to develop real life medicine. So the first one came out in 1999, which was a viral infection stopping drug called Formiversin. And till today, like 2019, you have almost 9 or 10 drugs, and they administered in a different forms. Like some of them are injection in subcutaneous, some of them are your intravenous, some of them are intrathecal. So different types of injection or administration of drugs happens, and you get good result, or you can actually knock out or knock down certain genes. And the last slide actually tells you all the drugs which is available in the market and the companies who actually develop this type of drugs and the certain disease which is addressing by development of these drugs. So you can see some of the big names of United States like Ionis Pharmaceuticals, Genzyme, Biogen, Alnylam Pharmaceuticals, Sarepta Pharmaceuticals. They are all working on this genetic medicine development field or your gene silencing development field. And even I just put like 10 names which is exclusive very much for this particular type of technology. But obviously there are other gene editing companies in United States, like you heard of the name like the vaccine company called Moderna. They are also working on mRNA. Like there are like so many different biotech companies working in this particular gene related disease areas. So which is really exciting, and we hope like in next 10 years or so we'll be having more progress and there will be more drugs which basically really important for certain diseases which is not addressed yet. So I will stop here. I know there are a lot of informations, but if someone is having any questions, I will be happy to answer.
Thank you so much, sir, for your presentation. It was really great. And yeah, we have got some question. First question from Dr. Ashwini Kumar. Can DICR or similar enzymes be used as therapy in order to degrade the target mRNA?
Yes, I mean, just DICR. You probably heard of the name of called CRISPR-Cas9, which helps to edit a particular gene. So by principle, you can use different types of enzymes or proteins which helps to cleave the target mRNA. So there are companies who actually develop different types of proteins which helps to fine tune this type of technology. So by principle, it is possible, and that's why the gene editing companies, they use the CRISPR-Cas9, or there are so many different type of Cas protein which actually helps to edit or to silence particular. So which is probably some ongoing work. I mean, if you look at those companies' website, you will get like a better picture, like which company works in which technology.
Okay, sir. Then question from again Dr. Ashwini Kumar. With specific gene sequence as target, is it good to have trial in animals for this nucleic acid therapy? Can transgenics be reliable animal models?
Yeah, I mean, which is, I would say, that's why like it took so many years to develop one single drug, because what happens, you develop a drug in especially in your preclinical area where you actually run all these assay and all those animal models. If your model is not actually translating exactly same thing as humans, you won't achieve that particular efficacy in human models. So that's why even in some of the cases you get really great result for your animal model study or preclinical study. A lot of drug actually fails in your actual human trial because it cannot actually the same mechanism or same efficacy in the human body. So it's kind of a trial and error thing, but obviously that's why before it goes into the human trial, they actually try to get a best suitable model which actually kind of resembles very much the human model, and based on the result they actually further proceed. It's not that if you get a very good result in your first or second animal trial you send the compound to the human trial. It doesn't actually work that way. So it goes through different phases, and with all these phases, if they are very much certain, then only it actually sends to the human trial.
Okay, sir. Thank you so much for giving us your valuable time. Thank you, sir.
Thank you very much for.
So now I would like to request respected Dr. Porimal Sarkar, Head of the Department of Physics, Cooch Behar College, to give vote of thanks to our honorable resource persons, guests and participants of the webinar. Please, sir.
Am I audible?
Yes, sir.
Savanti?
Yes, sir.
Thank you, Savanti Ghosh, an organizer, to give me an opportunity to give the respect and vote of thanks to all. Respected audience, I would like to thank to all the persons such as organizers, honorable research persons, dignitaries, guests, about 210 registered participants from the states of West Bengal, Delhi, Tamil Nadu, Karnataka, Uttar Pradesh, Tripura, Chhattisgarh, Assam, Jammu and Kashmir, Rajasthan, Sikkim, Goa, Punjab, Jharkhand, Haryana, Maharashtra in India, and from countries like Pakistan, United States of America, Nepal, Nigeria, associated with One Day International Year, thereby making the seminar really an international in character on drug discovery, development and vaccination, organized by the Department of Chemistry, Cooch Behar College. First of all, on behalf of my Department of Physics, Cooch Behar College, I am grateful to thanks Dr. Ravindra De, HOD, Chemistry Department and convener of this program, for welcome speech. He has welcomed all and he has explained the relevance of webinar during COVID-19 pandemics. I have the honor to thank Dr. Goutam Biswas sir, Honorable Vice Chancellor, Cooch Behar Panchanabanga University, for his nice speech as chief patron. He explained the relevance of webinar, chemistry, biochemistry, importance of vaccination, history of discovery and different vaccinations. They are taking long time to come to the humanity. He explained the role of government, private companies in interference to discover the medicines. And then next, I like to thank Dr. Abdul Kader Safili sir, Register, CBPBU. He was absent, but his blessings are with us. Then I would like to thank Dr. Pankaj Kumar sir, Principal, Cooch Behar College, for his speech as chairman. We welcome all related persons and hoping the success of the seminar. Then I would like to thank Dr. Kumar sir, Dean, Faculty of Science, Technology and Vocational Studies, Cooch Behar Panchanabanga University, for keynote speech. He mentioned the thanks for invitation, organizing the webinar by Cooch Behar College. He told the necessity of it is the mother of all inventions. He wishes the great success of the webinar. Now I would like to thank all the research persons. First to Dr. Anamitra Chatterjee, Regulatory Affairs Associate, Novartis, Slovenia, for his valuable talk in the first plenary session. His journey is starting from Bakura to Delhi, IIT Bombay, then Switzerland to Norway, Germany, then Slovenia. Now he is with us today. His field is organic chemistry, pharma industry. His topic was drug development. Secondly, to Dr. Ashwini Kumar, Research Associate, IIT Delhi, India, for his talk in second plenary session. His journey is starting from native of Bihar to IIT University Noida, National Technology Kerala to NIT Raipur to IIT Delhi. His delivered topic was immunity and vaccines. Thirdly, to Dr. Goutam Biswas, Assistant Professor of Chemistry, Cooch Behar Panchanabanga University, India, in the third plenary session. His journey starting from RK Mission Residential College, Nandapur, India Institute of Chemical Biology, Kolkata, then to CBPBU University. The topic of his speech was nanomaterials, a possible therapeutics in SARS-CoV. Fourthly, to Dr. Arindom Chatterjee, Scientist, Web Life Sciences, Boston, USA, during today's fourth plenary session. His journey starting from Ashoknagar, North Biswas, West Bengal, to RK Mission, Vidya Mandir, Delhi Mott to IIT Madras, then Jawaharlal Nehru Center for Advanced Scientific Research, Bangalore, from there to University of Connecticut, USA, and then to Web Life Sciences, Lexington, USA. He has nicely delivered lectures on the topic gene silencing in drug. Now, I would like to express my special thanks to Dr. Shyasi Bhakto, Joint Convener of tonight's webinar, and to Savanti Ghosh for nicely presenting, anchoring, and conducting the webinar. And thanks to other members of organizing committee, such as Professor Somnath Saha, Head of the Department of Computer Science, Sri Arindom Chatterjee, SMT Subarna Panchanan, Department of Chemistry, Cooch Behar College. Last of all, a lot of thanks to all associated persons of Cooch Behar College. This college is such a college which acts always as a family in such an in each and every activity to make it successful. This one day on international webinar on drug discovery, development and vaccination. Just wait 2 months for COVID-19 preventive vaccine. Stay home, stay safe. Thanks. Thanks to all.
Okay, thank you very much, sir, and thank you everyone for being with us, and thank you for your cooperation. That's all for today. You will receive your e-certificate for participation by 20th September only after submission of the feedback form properly, which will be activated for 24 hours. If you do not receive your e-certificate by 20th September, then only you can email us after that day. And you will find an optional quiz on the webinar discussion in the feedback form. Please try to participate as we will select top 3 winners and it will be announced by mail. So thank you again. Stay safe and good night to all. হ্যাঁ স্যার বলুন। হ্যাঁ স্যার, না আমার খুব টেনশন ছিল স্যার, এত বড় বড় সবাই আর কি থাকবে বাইরে থেকে। হ্যাঁ স্যার, থ্যাংক ইউ আপনাকেও এই যে এরকম সুন্দর একটা অর্গানাইজ করার জন্য।